National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Divisi Engineering Pura Group didukung oleh Departemen Research and Development memproduksi mesin-mesin khusus untuk divisi-divisi produksi Pura Group. Melalui penelitian dan pengembangan yang berkelanjutan, divisi ini juga telah memproduksi berbagai mesin untuk industri pertanian. pengering gabah dan biji-bijian,
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences.
We profoundly request to all online participants to mute your microphone during the symposium. And we are highly appreciating if you are willing to turn on your video during this event. Thank you. For online participants, please provide your name using your unique ID as listed in the program books. For example, for oral speaker, you can write E7 underscore your name. For example, E7 underscore Nico. And for participant only, you can write participant underscore your name underscore affiliation. For example, participant underscore Nico underscore BRIN.
For information and also, all material regarding this event will be provided in the checkbox for the online participants. Meanwhile, from the offline participants, the program books is also already available. And please feel free also to download through the barcode in the program book. The certificate will be given to all participants who attend full session, either offline or online. So please fill the attendance list through the link that will be provided by the organizing committee. For the participant offline, we will have in the attendance list in the break room. And for online participant, we will provide the attendance list from the chat box. Today, we also have a quiz. So be prepared and stay tuned during this event. And before the closing session today, we would like to announce the best presenter for this, the best participants, and as well as the best papers. So make sure that you join with us until the end of this event.
Distinguished ladies and gentlemen, before we proceed to welcoming speech, apologize for the next session keynote speakers. We would like to listen and sing the national anthem, Indonesia Raya. All of the participants in the main room, please stand up. All of the participants, please be seated.
On behalf of the organizing committee, good morning and warm welcome to the
7th International Symposium on Green Technology for Value Chain 2023. Unlocking a sustainable future through green technology innovation. This event is proudly presented to you by the Research Center for Environmental and Clean Technology, National Research Innovation Agency, Republic of Indonesia.
I am Swasmi Purwajanti.
I'm Ardi Septian.
We will be served as MCs for today's event.
This event is also sponsored by PT Berka Niaga Medica, PT Horiba Indonesia, PT Maja Bintang Indonesia, PT Galik Binamada,
PT Siklus Mutiara Nusantara, PT Pura Barutama, PT Insani Energi Utama, PT Wiralab Analytica Solusindo, and PT Teknolek. We would like to say thank you for supporting our event this year.
Ladies and gentlemen, for our schedule today, we will have 5 keynote speakers and 5 oral sessions in parallel. Each session will be guided by the moderator. Before we begin our first talk, again, we profoundly request to all online participants to mute your microphone during this session. If you have any question for offline participants, please raise your hand before asking the question, and the chairperson will give you the permission to ask the question. And for online participants, please write down your question in English in the chat box during the presentation, or simply click raise hand button.
Okay, today we will have the honorable keynote speakers, Dr. Syamila Azman from the University of Technology Malaysia, Professor Samin Jinadasa from University of Perania, Sri Lanka, Dr. Nasim Amrelian from the University of Queensland, Australia, Professor Ji Chen Liu from National Taiwan University of Science and Technology, Taiwan. Mr. Ferdian Kusnadi, the country sales manager PT Berkaniaga Medica Indonesia. And our distinguished guests, presenters, ladies and gentlemen.
Ladies and gentlemen, to begin with, I would like to invite Leonard Wijaya, PhD, who will guide us as a chair for this first keynote session in second day of conference. Dr. Wijaya is recently joining the Environmental Nanotechnology Research Group. He received his PhD from Botany King University, Saudi Arabia. And we will also have the first session of these keynote speakers, Dr. Samila Azman from University of Technology Malaysia, and Professor Shamin Jina Dasa from University of Peradeniya, Sri Lanka. Dr. Wijaya, time is yours.
Thank you, MC, for the kind introduction. Ladies and gentlemen, esteemed guests and participants, it is my great honor to introduce our distinguished keynote speaker for this conference, Dr. Shamila Azman from the University of Technology, Malaysia. Please join me in extending a warm welcome to Dr. Shamila Azman as she takes the stage. Please, Doctor. Please. Okay. Dr. Shamila Azman completed her PhD in environmental chemistry from the University of Technology Malaysia and embarked on her career as a lecturer in the Department of Water and Environmental Engineering at UTM and led the department as the head department from 2018 to 2022. Her dedication is evident in her active involvement in various environmental research fields, focusing particularly on water quality monitoring and microplastic. As a consultant, Dr. Shamila has played a crucial role, notably as an environmental performance index consultant with the Ministry of Natural Resources and Environment. Her impactful contribution include over 85 scientific publications, the supervision of 62 postgraduate students, and active membership in ANALYS and the International Water Association. Today, Dr. Shamila Azman will enlighten us on a pressing environmental issue in her presentation titled Microplastic, Tiny Emerging Underestimated Threats. Dr. Shamila Azman will share her finding on the challenges posed by microplastic and their abundance in Malaysian rivers. Please, Dr. Shamila Azman, the stage is yours for your presentation.
Assalamualaikum and a very good morning to all of the participants for the symposium today. Firstly, I would like to thank the organizing committee. It is an honorable for the invitation to the symposium. It is an honorable event for myself and I'm sure for all of you also. All right. Thank you, Mr. Chairman, Dr. Leonard, and also I would like to also talk about microplastic. Okay, what is actually microplastic? It is actually a very tiny emerging underestimated threat because we like to use plastic so much, but now we are slowly going to single-use plastic. But then this plastic has degraded over the years. Okay. So I'm from Faculty Department of Water and Environmental Engineering from Faculty of Civil Engineering, University of Technology, Malaysia. So what is actually plastic pollution? When you talk about plastic pollution, it is everywhere that you can see we have plastics. And plastics, the chronicles of plastics started long time ago. They started doing research on to produce plastic since 1736. Then in 1909, there was a plastic patented, which is called Bakelite. And after that, in 1937, we have many creation of new plastic. So part of it are polystyrene, nylon, LDPE, PU, Teflon, PET. And after that, we have we also have a plastic era in the 1980s. Up to now, we are still using plastics. Then the characteristic of plastic is it is low cost of production, lightweight, it is strong. That's why we like to use it. It's also durable because it can it is able to carry heavy stuff, and it can withstand the temperature and lots of things. Okay, sorry. This is actually footsteps. If you go to the seaside or go to the beach, animals will leave footsteps, but what man leave is not only carbon footprint, they also leave all this rubbish and garbage. And this is one of the sites that is for tourism, but you can see there are plastic bottles also left. Sometimes we can see baby diapers beside tourism spots. So that should change. So this is an example of plastic bottles accumulated in a wetland area, mangrove area, because the mangrove roots, they are able to filter off large plastics. So this is in Port Klang, Malaysia. And when we talk about plastic, some plastic do undergo biodegradation, but not all plastic are biodegradable. So we would not have problem with biodegradable plastics, but we have problem with photodegradation, where the plastic, after being exposed to sunlight, UV radiation, they will become brittle and start breaking up. So most plastic will photodegrade over time, and this is what we have, meaning that it will break up and leave small pieces of plastic. So if it's small, less than 5 mm, is considered as microplastic. So how fast does plastic break down in water? We do not know for sure. Currently, many researchers are doing studies, but then how long it takes for it to break into tiny particles, fragment called microplastic is not certain yet. But what really matters is that the plastic will eventually remain in the river or in the ocean. So at such small size, it can be harmful to aquatic life. So this is showing you how long it decomposes at sea. We can see that plastic bottle will take around 450 years. Plastic bags around 10 to 20 years. Diapers will take around 450 years also. And the fishing line or the fishing net is used will take longer, which is around 600 years. So this is an example of, this is an example of a plastic for instant noodle. They dig it up the ground, and last year they found this plastic. So if you look carefully, the expiry date is in 1988. So it's been a long time inside the ground, but it has still not degraded. Okay, so basically about every piece of plastic ever made is still exists today, meaning that all the plastic made from many, many years ago, it's still there in the environment. So there are many news report on the newspaper, all talking about microplastics. And these are the countries that These are the countries that are highest ocean plastic waste produce polluters. This is actually a data generated from a modeling study on stormwater from the drain that will eventually go to the sea. But it's quite alarming. You can see that we are among the 10 highest producer of ocean waste. And then after that come COVID-19. And we have many more all these PPE being used, protective equipment being used. So we estimated that there's increase in medical waste during the pandemic, increase in use and demand of single-use plastics because we were wrapping things over and over with plastic. So this is a problem. So you can see that these are the quite high numbers of estimated face masks being produced throughout the world, with China being the most estimated one. Okay, so how from plastic to microplastic? Actually, when we started this research, we were having problems. We do not have enough funding. So we thought that this is the best thing to do in environmental protection, to start to study microplastic, because it's used the least budget of from our research grants. So plastics, it's not moving. Okay, plastics pollution, there are many sizes and categories. We have plastic in the macro size, and when you say microplastic, the size is around from 5 to 1 mm. And we have two types of plastic, personal care products. Later, I will show you. Okay, so this is what I was talking about. The personal care product, where you use your cleanser or the facial scrub to remove off that cell, that skin cell. So they use microbeads in the personal care product so that you will have when you scrub off that skin will come out. Okay, so initially they use chemicals from microbeads, for example, polyethylene, PP, PET, PMMA. So what happened is that these microbeads will eventually go inside the water bodies. So, okay, so this is the examples of the cleansers that you use, the scrub that you might use. So they all contains the microbeads that is used to help in the scrubbing process and to have a more beautiful skin. So the idea is that not to use microbeads from polymers and whatnot. So why personal care products have it is that because the size is too small. Okay, when the size is too small, less than 5 mm, when it goes to the wastewater treatment plants, it is not being filtered or being treated. Okay, what happened is that then the microplastic will be left in the environment, in the aquatic life, in the water bodies. Then it can also attract and concentrate other pollutants on the plastic itself. So eventually it might also be ingested by animals and would also bioaccumulate. Okay, so what happened is that the impacts of microplastic is that it is changing the properties of beaches. If you take some of the sand, it might also consist of microplastic. It might be ingested by animals. It might be vectors for harmful pathogens, and it's also magnet for toxin. So there are also physical threat, but this is for larger plastics, entanglement, suffocation, ingestion. And this is what happens. There are also studies that found microplastic in zooplankton, in green mussel, in birds. Some of the birds went plastic in an albatross. So what happened is that, of course, they still eat the squid, everything inside the body of the bird. Okay, so when we go to microplastic in water bodies, So microplastic, like I said earlier, there are two types. One is the primary microplastic. The other one is the secondary microplastic. It will then fragmentize, then with weathering or degradation or from photo degradation, and it will produce small size of plastic. Okay, so when you talk about primary microplastic route, so what happens here is that we have the microbits coming from the personal care product being used by you and your family, and also me and my family. So what happened? It transfers through the sink and goes, it will be unfiltered, goes through the sewerage system. Then eventually it will end at the river or at the sea. And the microbits then can end up in the sea and our food chain. So these are the characteristics of microplastics found in water body. There are many colors, pink, purple, dark blue, dark green, transparent. So there are also many types and also many shapes. Either is irregular shape of particles, thin, plain polymer, or sponge-like polymer. So we have many types of microplastics. And these are ocean plastic that come from the largest emitting rivers. So they make studies and prediction from the river, it goes to the sea. And if you can see from Klang, Malaysia is around 1.33%. And the fate of microplastic from land, river to estuaries and the ocean. So we had studies on the river that eventually 80% of the plastic trash in the ocean are derived from terrestrial activities. It all comes from the land and using the river as a medium route. And from 103 polluted rivers are all located in Asia, 67% of global annual input in Asia, and also only one polluted river has been reported in the European countries. So the river, the environmental issues is its abundance and fate of the plastics. Then it is being transported in the river, then it ends up at the sea. So this is what we are talking about. Larger fish might take macro size of plastics, and smaller ones will take micro and nano size of microplastics. And microplastic density in coastal area globally, we can see that the highest selected microplastic density were around India, Southeast Asia, and the Mediterranean. And there are also countries that have already banned using of microbits in their personal care product. For instance, United States of America, Canada, Italy, and also Britain. So they ban the usage of microbits in personal care products. And this is a case study that my student carried out at Sungai Skudai and Sungai Tebrau, which is in the state of Johor. And we find that there are microplastic from the surface sediment. You can see that it is actually under microscope, can only be seen under microscope because it's quite small. We have a microbit, a small one at the bottom there. The round one, this is the microbit. And then we have a film, fragment. It's quite colorful. And we found out that certain fish will eat certain color because it might look like their prey. And this is microplastic from the surface sediment studied. We have quite a large number from Tebrau River compared to Skudai River. And the factors of abundance could also be due to rainfall. When we studied that if there's a heavy rainfall, there's always surface runoff, then there's also higher intensity of microplastic during that season. And this is a percentage of developed and high built-up areas because when we collected the plastic in Skudai, it's considered as also an urban area and also the same as the Brow River. And after that, we studied microplastic in organism, in water bodies, and meaning that we studied the ones in the fish, the one in the vertebrates. Some researchers are studying microplastic in turtles, in even the turtle eggs, and some are studying in seabirds. So factors of microplastic ingestion by organism could be due to their feeding strategies or foraging behavior, meaning that it might look like their food. The color is like their prey, small fish or something like that. So it might be ingested by the aquatic life. And this is also a case study from Skudai and the Brow River, where we have African catfish that we dissected the GI tract and we found microplastics. And these are the microplastic found. There's fiber in different fish species, foam, film from tilapia, fragment from tilapia. So we have most of these in the freshwater fish. And the problem is that when there's digestive system blockage, what will happen to the fish? There'll be behavioral change, there'll be stress, there'll be growth retard, and many other problems. So effect of microplastic on ingestion can either be chemically, where we have the vector effect with contaminants, or it can also affect through physically. This is another study that we did that to see the effect of microplastic to absorb contaminants. We are studying the effects on heavy metals currently, where we put the microplastic inside the river and we see how much heavy metal is accumulated on the microplastics. And we also did microplastic contaminant absorption, many previous study. And there's also study vector through ingestion. So this is a study where my student did it with, Okay, what happened is that what we did was that we accumulated the microbeads in an aquarium tank, and we studied the effect of heavy metal on the microbeads and also how it affects the fish. Then there are many also effects of microplastic onto the sea bass. What happened is that we did a study on cadmium, chromium, and plumbum in the lab. Okay, so what happened is that we did acclimatization, then depuration, and after that we did an injection experiment where we put microbeads inside the aquatic inside the tank, and we sample every 24 hours. After that we dissect and dissect the gills, the GI tract, and also the skin, and see the heavy metals in the tissues. Okay, so this was an experiment conducted in the experimental tank. And the findings found out that most of the heavy metal ion detected in the skin compared to the gills or compared to the GI tract. So most of the heavy metal will be absorbed to the fish skin. And we also did study on in muscle. So there are many studies around the world that studied microplastic in bivalves, and this is our study. Okay, this is our study microplastic in mollusks. We call it green mussel. I think in Indonesia it's called kupang hijau. In my country it's called kertau, is it? In Malaysia it's called kupang. Okay, so what we did was that we studied a few a few type of aquaculture method. Either we use the raft culture, or use the long line, or use the smart system. And also we collect wild mussels to see which one would contribute to more microplastic in the system. So the work is a bit messy because we have to digest the tissue. Yes. Okay. Because we have to digest the tissue, or this is much better than because we have to digest the tissue, then we also have to
Yes.
okay. We can see that it's okay. Boleh? Okay. We have to do digestion, then after that, it has to be clean, then have to be looked under the microscope before we can determine what type of microplastic. And this is the procedure we have that we conducted. Then this is the study areas, Johor, where I come from, which is in the border to Singapore, okay, just across the strait. And this is our findings, that we find that station 5 with the long line system had 20% higher pump, and also the smart system had the lowest microplastic. Whereas if you look here, station 6 and station 7 is where the wild mussel is being caught. But it's also quite high, and it's a microplastic found in the green mussel. So because of the way the mussel is clustered with nylon, long line, so we plastic together, so that's why it can also accumulate in the green mussel. We also did study on loctites, Kelantan, so we still find all these type of fragment, fiber, many types of fibers. And polystyrene and yes. And we also did studies on siput bongbong. This one I'm not sure what it's called here. The strongest dog punch. We find also it's a delicacy in Johor. So basically, there are many studies that study the effect of human exposure and impact of microplastic. There were also studies that found that there were microplastics in the blood cycle, in the bloodstream. There are microplastics in the lungs. Okay. So there are also studies about microplastics. And one of that from 2000 to 2030. And all species that ingested microplastics showed a positive correlation between the ingested microplastic and physiological traits. There's even a ability of the polyethylene microbeads got heavy metals which finally would transfer to the skin. Other metals for chickens, and we can easily ingest microplastic in our food because we might not be able to see it because it's quite small. 5 mm, it will pass through your body. Okay, so they have already been detected in not only Asian food, but also in salt, sugar, and so on. So we can get to what we eat and so that we are not. That's all. Thank you.
Okay, thank you, Dr. Azman, for that insightful presentation. Now we will move on to the question and answer session. I would like to welcome the question from the audience for Dr. Azman. Yes, a moment.
Hold on, Ibu.
Yes.
Thank you.
Is it still not working?
So my question is about, is there any research team in your university in UTM doing the research on the microplastic remediation?
Okay.
So maybe next time we can collaborate for the research, especially for the remediation of the microplastic, because the data of the microplastic pollution is available mostly on the paper, but not many data about the remediation technology available today. So it's very interesting, and I think it's very urgent to do the research about the remediation in the future.
Okay, thank you for the question.
Thank you.
Currently, we have only started looking into literatures of doing the bioremediation because we already have the data on microplastics, right? I think many researchers, either from all Southeast Asian countries, have data. Indonesia have data on microplastic, Malaysia, Vietnam. And currently, remediation, we have not yet been doing it, but we started having one student who might look into it. Of course, remediation, there are other prestigious university in Australia that are working on bioremediation. Initially, what I said was that it is too small or too tiny that we cannot do anything. It's already inside the ocean. We cannot dislodge the ocean sediment. But now there are study in the lab where they study to see that the microplastic that has been at the sediment is being floated up, and then you can skim up the microplastic, but it is still in the stages at lab work. But Ibu, of course, we can do collaboration further on. Yes. 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 But the one currently being developed in Australia is they are using chemicals to make the microplastic float. Yes. Yes, yes. Yes.
Okay.
Oh.
For the second question from the audience, Dr. Nida.
Assalamualaikum, Dr. Sabila.
Yeah, Waalaikumsalam.
Dr. Samila, is very interesting your presentation, but I want to ask about this topic. The secondary microplastic are formed through the degradation process of plastic. Okay, the secondary microplastic can be degraded. Okay, currently microplastic are considered hazard as they can accumulate in the body. However, on the other hand, microplastic have been detected in feces. When I read literature, it can be detected in feces.
Oh, yes.
Okay. This question is, what makes microplastic harmful? Is it due to the plastic polymer itself, since plastic polymer are largely composed of hydrocarbon? The presence of plastic additive, or the ability of microplastic to absorb or to bind with heavy metal. And the second is, has there been a clinic study on this? The microplastic is harmful. Thank you very much.
Thank you, Ibu. Okay, there are research being done that they find microplastic in feces, either from human and also animals. Okay, how does this microplastic become harmful to our body? We are still studying it because we can only, in Malaysia, we can currently studying on organism like either is in aquatic life or in like chicken liver or something like that. Okay, but then how does microplastic become toxic to human? Okay, because it's just plastic, like we say, because it will not be that harmful if we touch the plastic. It does not give out any toxin or toxicity. Okay, but then we have to remember that this microplastic is small. Then it can also absorb all the contaminants if it's in the water body. Okay, for instance, if they are, let's say, zinc or aluminium, okay, then the microplastic can become a vector and absorb all the toxins on the microplastic itself. So there are also studies that show that microplastic can also absorb pathogens. So that is why it might be harmful. So let's say we eat microplastic. We have to be careful on which part that we eat. Let's say if the microplastic has toxin on its body, what we studied was that it was able to also absorb heavy metal and transfer it to the fish. Okay, but then with human, sometimes when we eat fish, we will cut out the gastrointestinal tract. Perut ikan tu dibuang. Okay, but there are also microplastics that are very small that it can block your, if it's accumulate, it can also block your GI tract. So it might be on blockage or it might be on vectors. What we studied was that it is able to absorb, become vector and able to absorb toxin. So that is where that it might matter in pollution sense. Have I answered your question? Thank you.
Okay, thank you. Is there any question from the Zoom participant? Okay, from Dr. Muhammad Nasir, please.
Okay, thank you, Dr. Leo. It's very interesting lecture from you,
It's
Professor.
still
Can
the
you
sound
hear
is
me?
still off, Doctor.
There's no
Can you hear
volume.
me? Okay, can you hear me?
Yes, just a moment, Doctor.
There's no.
Dr. Nasir, it's still off. The sound is
Okay.
still off.
Dr. Muhammad Nasir, we cannot hear you. Are you muted?
No, it's not muted.
Ah, okay. I think there's like a sound there.
Okay.
Now I can read.
Hello.
Yes, yes.
Hello, yes, we can hear you.
Okay, Professor, it's a very interesting topic for us. We also see how in our research we study mechanism of how the micro and nanoplastic will occur. So I will ask you, have you can you do the how to treat the microplastic in the water?
Okay, doctor. All right. How do we treat microplastic in water? Initially, many years ago, I said it was impossible because the microplastic is already in the water. Okay, but now we have studies doing how to treat the microplastic in water. Either you take out the sediment and we take out the sediment and we would be able to separate the microplastic. But now they are coming with chemical treatments that you can float up the microplastic, but it is still stages in the lab. They are still working in the lab to see how to treat the microplastic, either inside the river or inside the sea, inside the ocean, because we have lots of amount of microplastic. So there are a few universities working on it, but it is still not being done on field. It's still being done in the lab. Okay. But in my university, currently we're still waiting for funding to work on the treatment of microplastics.
Okay, I think it's very interesting, Professor. Maybe we can sing together how to treat the microplastic in the water.
Yes, yes, for sure. Thank you.
Okay, thank you.
Okay, thank you, Dr. Nasir. Is there any more question from the Zoom?
Yes,
No?
Mr. Chairman.
Oh, yes, of course. Professor Nusa.
Thank you for the interesting presentation. You said in your slide that you are what you eat. In the slide you present. So if there are microplastics in salt, in bottled water, like this is without a trademark, yeah?
Okay.
Yeah. What kind polymers do you think the best packaging material, polymer packaging material, yeah, to change the plastics as packaging material? What kind polymer is the best? Because the use of plastics is all over.
Understood.
Especially for the food and drinks. Thank you.
Okay. Thank you, Prof, for the question. It's quite difficult to answer because when we talk about plastic is cheap. Everybody wants to use plastic. Like you said, it's everywhere we're using plastic. We are also using plastic not only let's say not only for water bottles, we are using plastic for food wrapper. Okay, then there are now scientists working on like there's like this bubble of things that you if you want to drink water, it's like I think a piece of like a membrane or something that the water is in a bubble, but it is quite expensive. So that is the issue now. Oh, what kind of
Polymers. That's
Okay.
what I mean. What kind of polymers could we
polymers to be
do?
used.
Yeah, to be used to
To build the
to
plastic.
be the bottles, cans, and also to wrap the food, yeah, as food packaging.
Quite tough.
Okay.
Quite tough question. Okay, because for the packaging, what type of polymer? I do not have the answer. But for food packaging, we are coming back to using the paper packaging, right? But for water bottles, it's very difficult. Okay, but I'm so sorry, I do not have the type of the polymer to use. But I did see it on a documentary where they're using the there's a round thing, okay, that you can also drink to quench your thirst together with the wrapper. Yes, but I don't actually remember the name of the polymer. Thank you.
Thank you, Dr. Nusda. If there is if there is no more question, I will summarize this session. Is there one more question?
We have.
Do you still have time?
Okay, thanks for the interesting topic. First of all, my name is Sumu Kulsum. I'm from 10 November Institute of Technology. In this opportunity, I would like to ask two questions. For the first question, which one is more effective to reduce microplastic contaminant between biological remediation or chemical method or using functional materials? And for the second question, how long it takes time for biological remediation for treat microplastic? Thanks for the opportunity.
Okay, thank you for the questions. More to bioremediation, meaning how long, if I got it correct, is that which one is better, either chemical or biological treatment, right?
Yeah.
Which method is better, biological or chemical treatment? We are still doing the process, still in the research part. So they have not come up with what is the best method. But I think what would be best if we stop using single-use plastic, whereas instead of using these plastic bottles, we use the reusable container for our daily life. Okay, so that's why in certain airports we have more of this water system that you can just take your bottle and fill up your own reusable bottle. So meaning that we go to single-use plastic. The second question is about I actually forgot. The second question is which one is the best, is it?
And the second question is how long will it take to dig?
Oh, how long? No answer yet, because meaning the process of how long the treatment will happen or how long it takes to degrade biologically, is it?
Yes.
Okay, so how long? Still there's no answer because it's still trial and error.
Okay.
Okay.
Thank you. So the time is limited. Before we close this session, I would like to invite Dr. Syamil Azman for a photo session and to present the certificate of appreciation.
Oh, okay.
Hey, thank you, Dr. Azman, for the nice presentation. Now you may return to your seat. Is Professor Jina Dasa ready for the second part?
Hello.
Okay, our second keynote speaker is no less distinguished. We have the privilege of hosting Professor Syamin Jinadasa, a professor in the Department of Civil Engineering at the University of Peradeniya, Sri Lanka. Professor Jinadasa received his PhD in Environmental Engineering from the Graduate School of Science and Engineering in Saitama University, Japan, with a dissertation title Constructed Wetland for Sustainable Water Quality Management in Tropical Region. In addition to his role as a Fulbright visiting scholar, Professor Jinadasa brings extensive expertise in water quality management and community engagement. With a career that spans from being a research assistant at the National Institute of Fundamental Studies to becoming a senior lecturer at the Department of Civil Engineering, Professor Jinadasa has dedicated himself to addressing water-related challenges. His expertise encompasses water treatment and reuse, water conservation, and efficiency. Today, Professor Syamin Jinadasa will be sharing his insight on water quality management and community engagement. Let us extend a warm welcome to Professor Syamin Jinadasa as he takes the stage to enlighten us on the crucial intersection of water quality management and community engagement.
Thank you. Hope you can hear me and see the screen.
Yes, we can hear you, Doctor.
Sure, thank you. So first of all, I would like to thank the organizers for inviting me for this presentation, and especially Dr. Nani, who was working with me and who is having a long collaboration. So thank you, Dr. Nani, for inviting me and arranging. So first of all, actually, I am from University of Peradeniya, Sri Lanka, and also I have some affiliations with the USA and other countries with the universities. So that maybe if I tell you a little bit about Sri Lanka, maybe you could visit one day if you have not visited yet. So it's a tropical country with the temperatures range from the 15 to 30 degrees. And we have in Kandy, we have around 2,000 millimeter annual precipitation and good water resources and also and nature-based systems. So that we have the World Heritage some places here, and in Kandy, the Temple of Truth, Sigiriya, and also we have this spicy food similar to like Indonesia, and also my university. It's called Garden University and multidisciplinary faculty. It's one of the most beautiful universities in the world. So again, you are welcome to our university, and it's a university in the undergraduate and also some postgraduate and also research-based programs running, delivering the support to the education system in Sri Lankan government and the communities. So let's see if I come to the point that what are the challenges we face. Actually, in the cities, we don't have much space for wastewater treatment, and also lots of vehicles moving here and there, traffic and water pollution we have seen in the urban areas, and limited wastewater treatment plants, so centralized plants, In addition, when you go to little away from the cities or the agriculture sector, you have the agriculture, the pesticides and other agrochemical usage, as well as the some kind of systems we have to depend on this fecal sludge management systems when you have the decentralized system, so limited capacity for septage management. So these are some of the challenges we face currently, and also we don't have we don't have a big issue with the drinking water, but still we have the enough amount of water or quantity, but some places like 10 to 15% of the population know the lack of the quality water. When you go to the discuss the sanitation issues with the little away from the urban areas, so these are some of the areas that people live in, we call slums, or we call estate sector. So that these toilets are located very close to the drains, and the people, they use the water, and their waste or the used water goes just into the water bodies, and this you see some of the discharge into the natural surface like that. And in addition, we have another issue with the hospital wastewater discharges. Our hospitals, this is my town in Kandy, we have several hospitals located in the town and also along this catchment, so that we don't have a good individual or dedicated wastewater treatment facilities for the hospitals. So now we have a centralized plant recently established, but not all the hospitals are connected. So these hospitals or the wastewater, when we discharge to our water bodies, there will be several pharmaceutical compounds coming into with this treated water, so that it will affect our water sources or the drinking water sources. So these are kind of challenges that currently we face in the water sector. So when we see the try to understand a solution, or how to find a solution for a particular problem, we see this one can be like a breakdown. So we have several steps involved in this kind of process. So what we have to do, first identify the problem very clearly. So then we may need to have some kind of research or kind of development strategy to test some of the research, and then once you have some of the research results findings, rather than keeping them in the laboratory, we have to deploy them, so that we have to go in the full scale. Also, without stopping or without just deploying in the field, we have to see whether the systems are sustainable, or how the operation and maintenance, whether this our whatever we implemented are being well operated and well maintained. So this is very important aspect. So as I mentioned, first is the science. First thing is first. So we have to have some good idea why it is happening. So what are the reasons behind that, and we need some scientific information. Then comes the engineering, like how to make this happen to the scale. Like we have some kind of research and development in the laboratory. So we do some kind of engineering to see how this as a system we can develop a solution. And the deployment, where we bring this into the implementation, where we need funds to implement as a full scale. And also, what is the final implementation model? Who will take the ownership of this system? What is the business model after this? So as you see, in like our countries, even some like in other developing countries, what I have seen is this fourth part, or sustainability. this part is very much lacking, and where we need to give a full attention when we have this kind of engineering interventions. So accordingly, I'll take you a few examples of what we have done during the past. So let's see that we have one issue called chronic kidney disease in Sri Lanka, where it was mostly affected in the dry zone of Sri Lanka. This is the dry zone. You can see I'm showing here the CKD patients where we have found, and it's the prevalence like 15%, and mostly the male farmers were affected by this. And these were research were carried out for a long time, and the WHO has found out that to recommend that with their findings, water as the one of the important factors where for this possible reason, as well as where we could intervene to help these communities. So this is where that we can see where we can bring the engineering and technology to help the communities. So when you see this dry zone of Sri Lanka, it's mostly covered by the groundwater, the supply, like maybe like 40% of the groundwater based drinking water supplies. So whatever the solution what we are going to bring to these communities will based on the groundwater, and also the technologies will based on the groundwater treatment. So if you see the Sri Lanka, the water quality or groundwater quality in the like overall, we have mostly there are 2-3 issues with this groundwater quality in the dry zone. One is that total hardness, where you will see the high hardness conditions in the this dry zone area, and the other one is the fluoride concentrations, where you see high fluoride concentrations in the dry zone conditions, some cases even higher than 8 milligrams per liter, and also some of the cases the nitrate concentrations, where you see here the first diagram like this Puttalam area, we have the high nitrate concentration. So accordingly, we did some kind of water quality evaluations, and kind of came up with the groundwater atlas. So this is actually our collaborators developed, Chinese Academy of Sciences developed this kind of findings, where if you see the dry zone, the like more than 50% area has exceeded the hardness levels, same as alkalinity, and fluoride concentration again around 10% exceeded the most recommended levels. Likewise, there are several water quality parameters that we have seen in certain places and exceeded the standards for the drinking water for the people in the dry zone. So based on this one, we came up with a project to see identify the safe, stable, and cost-effective membrane-based water purification systems for the CKDU affected areas. So we we have several steps on this process. First, we try to understand the water quality in the area where the CKDU affected area, and correlation between especially the dissolved organic carbon and the iron. So because this was the first time try to understand or characterize the organic carbon or dissolved organic carbon, as well as other metals, whether any interaction on this area. So same time, we came up with the two different methods. Usually in this area, most of the previous work and the government, they have used the reverse osmosis systems, but we came up with two new systems to investigate, considering their potential, the nanofiltration and electrodialysis systems. So that we compare this RO and even reverse osmosis and nanofiltration treatment systems, as well as we further studied whether how feasible these nanofiltration systems, whether it's technically, socially, economically kind of evaluation to see whether this is more suitable to apply for the dry zone conditions. So that, because what we have noted is, considering the dry zone water quality or the groundwater quality, the nanofiltration systems may be a better option, because most of the cases issue is with the hardness. So when you have certain places you have the fluoride concentrations, of course you need to have some kind of integrated systems with the nanofiltration. But otherwise, and considering the energy aspect, cost wise, and recovery aspects, we found that nanofiltration would be a more the better option. But mostly the important thing is, we have, implemented these reverse osmosis systems, other systems in these communities. But what has happened after 1 year, 2 years, these systems sometimes were abandoned, no connection with the government authorities or the water authorities, and they have not been used by the community. So what is the reason for this kind of thing? So this is where we think that the community engagement is very important for this kind of conditions. So when it comes to the nanofiltration systems, what we think, when we have design, when we come up with the proper design, we need to have a discussion with the community and decide where it should be located, like where we need to have some kind of guidelines for unit establishment and management, because before the establishment we need to have the concurrence from the communities and their willingness to support it. So then you need to have a technical support throughout the period, and also people should aware of the water quality and other risk factors. We have to do some kind of awareness programs, and also maintain regular operation and maintenance reports of the system, as well as the bringing this community, and ownership will be taken up by these communities and have a sustainable filtration system. So what I was trying to highlight here is, we have done more research, we have more technological solutions, we have been implemented these the filtration systems everywhere, but most of the things sometimes were not successful because lack of community participation or the awareness of the people. So that it's very important that these solutions are integrated with the communities, and take the ownership of these systems by the community. I'll highlight another aspect regarding the waste management, how this issue actually affect like our countries or the developing countries, because most of this now we are coming up with this 3R concept, 5R concept, and so and so. But still you see the some of the dump sites or the wastewater dumping throughout some places. So most of these dump sites are located unfortunately close to our water bodies or the rivers. So what will happen, whatever the leachate coming out, the watery part coming out from this solid waste or the dump site, it will leach, or it will go into our the water resources or the water bodies where we take the drinking water. So it's another issue, or we need to find out what are the possible solutions, so that accordingly we try to investigate what is the leachate characteristics of this in our country dump sites or the landfills, and see what kind of solutions or what kind of treatment methods are more suitable. So if you see our conditions, like mostly our leachate is, most of the places is very diluted because of we have the high rainfall situation. So if you see the BOD COD ratio, like some places we have the more than 0.5, but other places it's very less BOD or COD ratio. And considering that, we came up with the system to try the treatment where the membrane bioreactor system with the anaerobic, anoxic, and aerobic system, we call multi-stage treatment. to see whether we can manage or we can treat this leachate effectively. So we have found we can, our results have shown you can get a very good results, but you need to change the operating conditions, other aspects based on the leachate characteristics of the system. So based on the age of the landfill, whether they have done the separation or not. Likewise, considering all these aspects, we came up with these membrane bioreactor systems to treat the leachate, and the results were promising where it can be used by the government authorities. So when we consider this technology-wise, as I mentioned earlier, whatever the technology when we see, we have to bring as a appropriate technology where you have to consider the financial conditions, whether it's technical conditions, it's matching our country conditions, climatic conditions, and socio-economical, institutional, even cultural. So that this that's where we think this as appropriate technology. So these technologies has to be matched with all the conditions. If it is not matched, what will happen? It will not sustainable. So that these systems will be, installed, but it may not able to continue as it is, like what we planned. So this is where we see the issues with some of the solutions being implemented. What we have to do is always appropriate technology and local technology, and where it suits to country needs. Accordingly, we consider the constructed wetlands. So wetlands may be a good solution to improve the water quality in our country like Sri Lanka and tropical conditions. So accordingly, we have tested these wetlands for few occasions. One is that as the floating wetlands we have implemented in the Candy Lake. So it was very successful with these flowering plants. And here you have seen in the small community, like grey water treatment, as I mentioned earlier, some of the communities, when you have don't have very good sanitation, or sometimes we call like open defecation or like that, so that these natural streams may carry fecal coliforms and other stuff, so that we may have to have some treatment before it comes to the water sources. And especially the different systems like subsurface flow and vertical and horizontal systems integration to remove nitrogen compounds in the constructed wetlands. And also it's very important to study the kinetics in the tropical conditions, because we always have an advantage of the tropical conditions where you see that the other countries you have the winter and this kind of senescent space, but like Sri Lanka and others tropical countries, you have this summer period always, so that you have the good plant growth where you can have a very good system. So that this further studies is required, like microbial analysis of these, see how the tropical systems working, and with some interventions like the water level fluctuations, especially the integrating systems. Like sometimes in the wetlands we have the emergent plants, submergent plants, like vertical flow systems, horizontal flow systems. So how you can integrate these systems and bring a most suitable or more effective solution. So it's a kind of need where we can bring. So I'll take you another example where this community engagement has been noted very important. I'll take this Candy City, where is my hometown. It's the UNESCO heritage site, and very much tourist attraction is there. And the Temple of Truth, Buddha's tooth, is kept in the Candy town. So what happened? This Candy town or the Candy Lake is located in the heart of the city, and this lake was polluted because of the wastewater discharge, and there was a big issue with the fish death of the Candy Lake. So we want to identify what is the reason for this pollution and what kind of interventions we can do. So we carried out some kind of social surveys, talk to the people and discuss with them what their opinion, and same time we do some hydrological or hydraulic modeling to understand how the water movements in the lake, and then also the water quality measurements. So this is what I mentioned, the preliminary studies to understand what is the current situation of the system. And then we came up with some solutions with the laboratory testing, like floating wetlands like this, and then we implement these systems in the full scale. So as I mentioned earlier, we have some kind of laboratory or R&D, and then we bring these kind of solutions and implement it in the final stage. So when it comes to the final stage, the floating wetlands at the Kandy Lake, that we have implemented, and we came up with the sewage treatment plant as a model treatment plant to the city, to protect the lake, where all the hotels, the tourist hotels, can use this as a model to implement. And also the awareness, formal discussion with officials and other organizations, and bring this kind of all the school children, other people together, and come up with the solution to mitigate the pollution in the Kandy Lake. So that was one example from the town Kandy, how we have managed the water quality. So I'll bring you a little bit more from the water. I started from the groundwater, and then to the water quality, and the other ways. Then now I just highlight about the wastewater conditions, and also especially the industrial wastewater, how it is important. So that when it comes to the industrial wastewater, I mean, in our countries, based on the other industries as well, the characteristics may different time to time because of these different operations and other stuff. Okay, and also the discharge standards. We don't sometimes they change time to time, and also with the suitable to the industry, and the appropriate technology, as I mentioned, React space allocation, reactor configuration. These are very important. This industrial wastewater, especially like our countries, they face mostly problem with the sludge, where we have currently only one incineration or the kind of destroy this kind of industrial sludge, but the industries have issue with the how manage the sludge. And now we focus always this resource recovery rather than we dispose. So with the we need to come up with the systems, and energy usage is another challenge with these industries, even especially for the wastewater treatment plant, how much energy they consume, and the capacity building. As I mentioned, how much our engineers in the industry, the technological people, whether they are aware on the solutions, how we can bring this as a eco industry, or we have very kind of environmental friendly systems in the industry, and accredited laboratories also kind of very important to see what is happening in these industries, and the cost. So that is including the operation and maintenance cost. So we did some kind of studies for the, especially again for the rubber wastewater, another challenge in the treatment. So again, as I mentioned earlier, the first we have to study the fundamental sciences of this character, so that we do the characteristics of this wastewater. We try to find out what kind of compounds they are, because as it found difficult to treat this, so that usually you have the anaerobic, anoxic aerobic system, something like membrane bioreactor system, and but here what we came up with the new is the pre-treatment, where this handle this kind of high COD value with the to have a good system. Accordingly, once we have this with the pre-treatment, we were able to achieve a good finally the good the treatment as again multi-stage treatment to combining these three different types like anaerobic, anoxic, oxic. Similar to the leachate systems, but here the difference is we need to have a good pre-treatment to manage the rubber wastewater conditions. And also the application of sequencing batch reactor systems, like even to treat the emerging contaminants, like oxidogenic systems in the anaerobic phase, as well as I mentioned earlier, to model plant in the temple, we used this system because in the temple we have especially the fluctuating flow conditions, and we have very limited space. So this system was very good to manage the water issues at the temple. But again, the important thing is to educate the temple staff or the communities, and then the ownership will be taken up by the plant of the treatment plant by the temple staff, and they maintain or they look after the temple, kind of thing. So I would like to come up to the final few points. Like one is the constructed wetlands could improve the water quality. As I mentioned, it's very good for the tropical conditions, and integrating the plant systems and plant management, understand the hydraulic conditions, like these are very important to have successful systems to improve the water quality. And when it comes to the other like water quality management in the lake or other place, integrated approach, both technology and community engagement was needed. And for the first time in Sri Lanka, we have done this kind of all the floating wetland SBR system and other systems. Maybe these technologies have implemented in other countries, but Sri Lanka as an integrated system with the community participation, and it was a very successful story, so that we implemented at the Handy Lake. And other issue I was mentioning, the groundwater treatment, like in the dry zone conditions, our research was like very much successful, bringing this nanofiltration technology as an alternative to the reverse osmosis systems, and we study thoroughly on the water quality, and also we bring them as a sustainable systems with the community engagement at the where we see as the successful story. And when it comes to the wastewater treatment, we have what we important is that the pre-treatment is a must for like this kind of rubber wastewater, and it has to be treated properly before it. And we have found this membrane bioreactor system or integrated MBR with AO anaerobic MBR and anoxic systems were very much successful to handle this kind of wastewater. In addition to the leachate, where as I mentioned, when it comes to the leachate characteristics, somewhat different from the rubber wastewater. So we create a different operating conditions, and then we were able to come up with the good results to manage the water quality. And especially this everything is bring end of the day with the community participation, and this involvement with the people is important to handle the solution. So finally, these work, our work, especially the link to our sustainable development goals, 17 goals, especially the clean water and sanitation, and the good health and well-being, and climate action and partnerships. So these are very important, like where it links to our studies, and we can see this all the aspects are linked. And actually, I was thinking in a different way that this water and sanitation centered sustainable development goals, so that where we see this all the most of the other SDGs are connected, or there's a relationship with this. So as you know that we are planning to achieve the equitable access to safe and affordable drinking water for all, and the equitable sanitation and hygiene, improve the water quality, sustainability of water, increase the sustainability of water use, okay, and the water resources management, integrated water resource management, and restore water related ecosystems, and also international cooperation. That's also very important. You can see the 6A to handle. This is where that we bring like our country, Indonesia, and different countries together to see what is how we handle these kind of situation jointly, and support and strengthen the local communities in improving the water and sanitation management. So that although it says given the last point 6B, but I think again we have to give more attention for the strength and the participation, support the local communities in improving the water and sanitation management. So while we have this all the technology, we have all the solutions. So this community engagement has been very important, and we have to link with this overall process. So again, thank you for inviting me. And this is some of the work I was planning to share with you. And if there's any question, I'm happy to answer.
Okay.
So, yeah, thanks.
Thank you, Professor Jinadasa, for that insightful presentation. As we move on to the Q&A session, I would like to welcome questions from the audience for Professor Jinadasa.
Since there's an
Maybe-
online question.
Okay, maybe online question first. If there is no question yet, I have a question. In your opinion, which pollutant caused the most damage to the 3.4 million people in Sri Lanka? And what have the government done on this matter?
Yeah, so the when you come to the water pollution, the we have the urban and rural areas. So when it comes to the urban areas, mostly as I mentioned, wastewater discharge into the natural or our water bodies is a major issue. When it comes to the rural areas or the other kind of areas, especially the groundwater is being used, so that when it use the groundwater, mostly the agriculture or the fertilizer usage, and sometimes in the natural conditions may occur, kind of some reason. So this is a kind of combined effects and different effects or different reasons for different areas. So what we have to see, rather than exaggerating the current situation, where we can stop this situation. So especially when it comes to the Kandy city, we came up with we call centralized wastewater treatment plant to the city. So, but what we have seen or what we were thinking, again, we were rethinking whether it was the best system to our city. So, because whether we should go for a decentralized system, whether that would have been better, kind of bringing the cluster systems, because when you implement a very similar system like in a developed country, so the developing country context or the environment is not the same as the other countries. So, for example, I tell you, when you dig the sewer line or the sewer pipes to install the sewer network, the once they dig this for a road, they have to repair the road. Then again, the water line comes, the drinking water supply. Then the telecom line or telecommunication. Then the Ceylon electricity line. So that, and also there's a problem with the cross contamination and this kind of the managing the system, so that we have to be very careful in selecting the technology for kind of especially for the developing country context, and go decide and how we can implement, and also how we can recover the cost of the solution, so that if you implement a solution, what is the way of recovering the cost? Like it can be a loan, it can be a kind of the aid. So end of the day, it's like crucial, like the Sri Lanka facing a currently economic crisis. So this is very important for us to see how you kind of recover the cost of the solutions that we implemented. We need to have again sustainable systems. Otherwise, if you implement, we as I shown you in some photo, that it's we invest huge amount and we have the system, but end of the day we cannot run the system. So these are the challenges that what we are facing, mostly about the kind of water in the urban and the rural sector.
Okay, thank you. And there is another question from Dr. Desak.
Thank you very much for the time. And thank you for your nice presentation. I would like to ask about the reactor that you use. Are you put the membrane inside your reactor, and how the function of microorganism, if microorganism can be to purify the water?
Yeah, so actually, I think we have used two different types for a few cases. One is the sequencing batch reactor system that I mentioned for the to manage the wastewater at the temple, and also to manage some kind of industrial wastewater, so that we maintain like all the microorganisms inside the reactor, and we run the system as a batch system. I mean, kind of batch system to handle the fluctuating conditions. When it comes to the membrane bioreactor systems, yes, we have put the membranes like as submerged in the membrane reactors, and we run the systems while we have the maintaining the MLBSS in the bioreactor system. So this some of the cases like anaerobic MBR we maintain as a submerged reactor, and some of the cases we run as the external reactor cases. So at this moment we are testing whether it's more suitable to as a submerged or as the external condition. So that is also another option that we are currently being studied. Yeah, thank you.
Yeah, thank you very much, Professor Samin. I'm Hanis from BRIN. I'm very interested in your presentation about the floating wetland system for the phytoremediation of the lake. My question is, what kind of the contaminants in the lake in Sri Lanka, and what kind of plants did you use during the phytoremediation on the lake, and what did you do with the plants after the plants fully accumulated the contaminants, especially maybe heavy metals in the lake? Thank you very much.
Yeah, thank you. Actually, these floating wetlands are being implemented when you come to the selection of plant species. We have done like some research. I showed you some slides that in the laboratory scale, and we tested for some kind of typha plants, and we have called scirpus, like scirpus roses, like the type of plants, macrophyte plants, and also cannas. We call cannas is a flowering plant. So actually, from the research we found that there's not much difference in these removal efficiencies, but the cannas was, I mean, some kind of not the best plant in removing removal efficiencies, but when we have the discussion with our irrigation department who manage the lake in the urban lake, they were interested. They have an idea. We came into agreement that canals can be used because of the integrate with the landscape, and the people may think on that. So because of that, we decided to go with canals with the moderate efficiency. Where nitrogen, phosphorus, and carbon removal, we did some laboratory and so on. Then when it's kind of the management, actually all these plants will come to the mature level. So in the tropical conditions, for sure, we have to do the harvesting, or we call it sometimes thinning, so that this plant management is very important. So time to time, you have to remove some plants, or you have to do the harvest. So this, especially the flowers, the harvested flowers, these flowers were taken to the school, and they have used inside the school for some kind of decorative purposes. And one more most important thing, regarding the contaminants in this urban lake, it is mostly coming from the hotels and sewage. So it's mostly it's issue with the nitrogen, phosphorus, and the carbon, not with the heavy metals. So that we were targeting these pollutants, and so see, especially these were implemented near the inlets, so that and also it's a kind of area we were trying to do a ponding, or I mean kind of we keep some kind of retention inside and then bring the water to the lake, rather than, like, for example, before the water coming into the lake, we have a kind of trap, like a seal trap, but this is a trap for the nutrients with the floating wetlands, and we see whether we can remove some of the pollutants. And the most important thing here is, I may be, if I ask you the question, if you see the floating wetlands with flowers, whether you liked it or not. If I ask you from you, if you see with flowers in the floating wetlands, it's lots of flowers in the lake, whether you liked it or not. Maybe you like it, right? So, but what I was trying to say is that actually, when you see this kind of flowers, or you see kind of high plant growth, that is a not good sign. That means you have high nutrient conditions in your water. So that this is used again as an indicator to see the pollutant level. So with the rate of plant growth and the rate of flowering time, so we investigate to what extent the pollution is, so that the people may like to see the flowers, but it's not in the reality. It is sometimes maybe a not good sign. But of course, if some plant, if you don't see, I mean, much of flowers, maybe a good sign in water quality aspect. It's good for the engineering intervention. So what I was trying to say, while we do the treatment, it is used as an indicator, as well as the people were asking in the community why you have installed this kind of system. So that people were trying to think, the walking around the lake, why these wetlands are being installed, why the floating wetlands are being installed. So that because it's polluted, so that people trying to discuss, take this message to their home, school children walking around the lake, they see these wetlands and they bring the message, see, the floating wetlands are installed because our lake is polluted. So this is very important for the next, bring this message to the next generation. That's where we bring the school children also into this community engagement. Yeah.
Okay, thank you, Professor and audience. Unfortunately, the time for Q&A is finished. Before we close, I would like to invite Professor Jinadasa for a photo session and to present the certificate of appreciation. Okay, just a moment. Okay. Thank you very much, Doctor. As we conclude this session, please join me in expressing our gratitude to both Dr. Syamila Azman and Professor Syamin Jinadasa for sharing their expertise and contributing to the intellectual richness of our conference. I apologize for any inconvenience that may have occurred. Thank you and see you in the next session.
Thank you.
Now I will return the program back to the Master of Ceremony. Thank you.
Thank you very much. Before we continue to the next schedule, which is coffee break, I'd like to ask Dr. Amanda and Dr. Sharmin and Dr. Wijaya to take a photo session. Would you like to come to the stage, please? Thank you very much. Yeah, ladies and gentlemen, it's time for us to have a little coffee break time, but because of the limited of the time, I'd like to request you, while you are doing coffee break, you may take maybe 5 minutes and then we come back to this room again, so we can have coffee break while starting the next keynote speaker. Thank you. And the coffee break will be on my right and left side. You can come back here at
10:15.
10:07. go back to here, and then you can bring your snacks while we are starting the next session. Thank you.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Research is our strength to overcome any limitations. Innovation is the guiding vision that drives us towards the future. We cannot face this alone. We must stand together to make our vision a reality. Growing strong upon the land of Nusantara, a magical place filled with miracle and wonder. A land spanning 2 continents and 2 oceans, rich in culture and biodiversity. We and Indonesia are the necessary combination to realize the grand visions of the future. Like an orchestra, BRIN harmonizes research, development, assessment, application, and also synergies, invention, and innovation in Indonesia. We created an ecosystem consisting of 7 deputies, 12 research organizations, and 85 research centers dispersed throughout Indonesia. Research Organization for Aeronautics and Space, Research Organization for Nuclear Energy, Research Organization for Energy and Manufacture, Research Organization for Earth Sciences and Maritime, Research Organization for Life Sciences and Environment, Research Organization for Electronics and Informatics, Research Organization for Social Sciences and Humanities, Research Organization for Archaeology, Language and Literature, Research Organization for Health, Research Organization for Nanotechnology and Material, Research Organization for Agriculture and Food, Research Organization for Governance, Economy and Community Welfare. BRIN catalyzing research and innovation to develop an advanced scientific ecosystem in Indonesia with two main targets. BRIN realizes the tremendous potential of the nation's youth in the field of science and technology. Thus, various research and innovation policies are being formulated to develop and cultivate human resources with high competence and aptitude in science and technology. Creating many excellent programs is a form of BRIN's commitment to researchers, innovators, and the general population from various disciplines across the nation. Research and innovation ecosystem quality improvement in Indonesia is also realized through the collaboration of various ministries, institutions, domestic industries, and regional governments, to name a few, by forming the Regional Research and Innovation Agency.
Sorry, you are mute.
Dr. Nasim Amiralian is the leader of the Bio-Inspired Material Research Group at the Australia Institute for Bioengineering and Nanotechnology, the University of Queensland, Australia. Her research focused on exploring the design and synthesis of tailor-made and precision engineered biomaterial using agricultural waste to create environmentally friendly product as a replacement for petroleum-based plastic. The outcome of her work has resulted in the establishment of Australia's first nanocellulose pilot production plant. In recognition of her contribution to the field of nanomaterial engineering and research excellence, she has received a number of awards, including the 8 Australian women who are seeking Up the Word of Science, Mary Claire 2020, one of Australian top 5 scientists, 2018, Queensland Women in STEM Prize Judges Choice Award, 2017, and Women in Technology Life Sciences, and our Infotech Rising Star Award, 2016. Dr. Amarlian also is a strong advocate for cultural diversity and equity, the support staff and students to grow as more effective leaders and create social good. Nanocellulose derived from non-wood resource will present by Dr. Amarlian. Dr. Amarlian, time is yours.
Thank you so much. I really appreciate the invitation, especially Dr. Amanda Septefani, a great former colleague of mine, and hold the conference organizer. It's my great pleasure to present some of our research data at this conference. Just checking that. Are you seeing my presentation? Presentation. Would you please confirm how do you see that? Do you see the full slide? Hello? Okay. Today I'm going to talk about nanocellulose derived from non-wood resources. So in my group, our activity is mainly focusing on the development of the sustainable material. We try to make innovative material and using agricultural waste, and based on that we do collaborate with the other colleagues at different universities around the world. We also, to do fundamental science, we also collaborate with industry in order to translate our science into the real world products, as well as have collaboration with indigenous partner, which I'm going to talk about that today. But first of all, I would like to acknowledge the team of these people who have worked with me over the few years, and without their contribution, this presentation and these research data wouldn't be possible. So giving you an overview about my group's research interests, I can say that we categorize into the 3 different areas. When we use nanocellulose as a template for the guided formation of nanoparticle or polymer brushes, for instance, for the conductive ink applications, or antimicrobial material or anti-corrosion coating material. We also use nanocellulose as a reinforcing agent, reinforcing a range of different polymers, reinforcing papers, or even making carbon fiber by adding these nanofibers into the polymers. We also use the whole agricultural waste and treat that and make bioplastic and sorbent, for instance, for water treatment. Based on that, I have a range of different kind of projects working with industries and some other research group to investigate the highly potential of these nanocellulose-based material. So today I'm going to talk about nanocellulose, giving a general kind of idea about what is nanocellulose and some of its application. One case study about Sphenifex nanocellulose enhanced composite, conductive inks, and water treatment. So we know that cellulose is the most abundant polymer in the earth. So we can find cellulose almost in any plant, but there are some other resources of cellulose as well. In addition to the wood or grasses, we have some alternative sources like bacterial cellulose, or tunicate. Some waste, agricultural waste, both processed and non-processed, they also have a great content of cellulose, which they are specifically my main area of interest. And I do believe that in Southeast Asia, you are also working on non-wood related biomass as well. So this is the hierarchical structure of cellulose. If, for instance, we want to look at any plant structure, they more or less have similar structure. So they made of fibres they attach to each other with different structural components like cellulose, hemicellulose, and lignin, which these are the three main components. If we try to just isolate these fibers and loosen the structure, isolate the fibrillate the structure, then we can have nanofibers or nanocrystal, which are the building blocks of any plant. So in the processing that we use for nanocellulose, it's not like wood, because grass or agricultural waste, they don't have very stiff and rigid structure like wood. So the process is just simpler. Basically, we take the ag waste, wash, after washing and grinding, a simple alkali treatment. And if we want, we do bleach and remove lignin from the structure, which we have more white fiber, which is more hydrophilic. But sometimes when we want to introduce some kind of hydrophobicity to the fibers, then we retain more lignin in the structure. As you see here in the graph, we are able to tune the lignin and hemicellulose different structural composition in the grass just by changing the alkali concentration in the delignification process. So each of these samples, basically, they give us different property profile, which is suitable for different applications. So for making nanocellulose, which is a general term, it contains both cellulose nanofiber and nanocrystal. We can do pre-treatment, specifically if we want to introduce a specific desired functionality. For instance, if we want to introduce carboxylic acid group to an negative charge, then we do the tempo treatment or carboxymethylation. But then mechanical shearing, it could be high energy bead milling, it could be high pressure homogenizer or ultrafiltration. Each of these kind of tools, they give us different types of fibers in terms of refining. They give us different level of refining of fiber. Then we end up to these thin and long, and you see that very flexible cellulose fiber. They have very high aspect ratio. The ratio between their lengths to width is really high, which is really good when we wanted to use them as a reinforcing agent for polymer. But we can also do acid hydrolysis in order to break down the structure to the short rice-shaped crystals. These are very stiff, high crystalline particles, and they are good for specific applications. So as I mentioned that we basically, especially in our cellulose fibers, that we don't do any intensive chemical treatment. Basically, we have a composite of fiber which contains hemicellulose and some level of lignin as well. So why nanocellulose? Because we know that it's green, is abundant, and it is a very sustainable and environmental source, especially when we are using ag waste. So we are adding value to our agricultural waste. So that's the reason, because of the importance of cellulose, it found great application in a range of different industry. It comes in different forms. It can be in form of dry powder or in the form of hydrogel or film or membrane. And they can be used in flexible electronic industry because they are transparent, and they can be used as a template for the introduction of the conductive material. They can be used in biomedical applications. They are biodegradable and they have a great mechanical property. Same with in other industry like food and agriculture, textile, water treatment, or nanocomposite, which is the most common one. So I would like now to just move back to talk about some research that I've done in the past few years. Basically, one research is based on Spinifex. Spinifex is Australian endemic grass. It covers 30% of Australian land masses. And before my PhD that I started to work on that, basically there was no scientific work on the Spinifex grass. So this despite we didn't have any information, this grass has been used by indigenous Australian people for more than a million of years. They have the knowledge to use this grass for different applications, for instance, making these basketry household items or shelters, or also some of these grasses, it has more than 120 different species. Some of these produce a sticky resin. That resin can be as a very strong adhesive material for mending cracks or making the hafting tools. So what I found that very thin and long cellulose fiber can be extracted from a spinifex grass using minimum mechanical energy or chemical treatment. The reason behind that, because this grass grows in a very hot and dry Australian desert environment, so it has to find a strategy in order to survive this kind of environment. That's the reason it has very loose structure. So based on that discovery, a range of different applications for spinifex grass was developed. We used that as a reinforcing agent for reinforcing cement, or as a wound dressing material, or reinforcing range of polymers, making a composite, as well as cardboard boxes, which I'm going to talk about some of that. And more importantly, this is something that I'm very proud of, that Australian first nanocellulose pilot plant developed in Australia, but in collaboration with our indigenous partner. The aim is that we have... Pardon? Basically, we have...
saya pikir, oh ya Pak, saya pikir di Unhas Pak.
Sorry.
Yeah.
We have two pilot... Sorry, can I continue? We have two pilot plants.
Yes, you can continue, Doctor.
Thank you so much. One is led and operated by our indigenous partner, which is providing job opportunity for indigenous training. So I would love to talk about that elastomer reinforced cellulose nanofiber. In this case, we use the spinifex. For instance, we look at the medical gloves. There is a huge market for medical gloves, especially after COVID. We've seen that the importance of using these increased as well. So, but the problem is that most of these medical gloves, they have a really good tensile strength. They are strong. They have a good toughness, but very low flexibility. Why we need that? For instance, imagine that the surgeon wants to do surgery. They need a thinner membrane, but at the same time, stronger and flexible in order to give the true feel of sensation. So far, the kind of existing approaches is reinforcing latex, natural rubber latex, with a range of different fillers, which most of them are like graphene, carbon nanotube, or silica, but they are good. They improve the tensile strength. And then, but the problem is that because these nanomaterial reinforcing agents are stiff, they increase the stiffness of membrane, while we wanted what is desired, increase the flexibility and increase the elongation and break while maintaining the high tensile strength. So our hypothesis for that one was that, okay, how about if we use soft and tough nanofibers in order to make the thin and tactile membrane? So to do that, we prepare a range of different types of cellulose nanofiber, all high aspect ratio nanofiber, which the difference between these fiber through different treatments, they have different hydrophobicity and surface functionality. And we benchmark it against the commercially available cellulose derived from wood. So as you see that in this case, when this is just sodium hydroxide, it has some lignin in the composition, which is in comparison to this one, which is bleached and lignin is removed, is more hydrophobic. And while this is the commercially available one, is very hydrophilic. And this is the sample, which is treated with dieutectic solvent, choline chloride mixture of choline chloride and urea, that has the highest hydrophobicity. And this is the contact angle value of the natural rubber. So what we see that with addition of very small amount of only below 0.5% of these more hydrophobic cellulose nanofiber, we increase the tensile strengths while we, without any compromise in the, actually in this region, in their tensile strength. So we found that hydrophobicity is a key, so lignin helps us. The next step, we're thinking how we can increase the content of lignin. As you see here, I earlier mentioned that with changing the concentration of sodium hydroxide that we are using, we can tune the lignin content. So here it's showing that with different concentration of the sodium hydroxide, we have different contact angle value. This was the original work that we did, and now we've been able to increase the contact angle to 69 with using only 0.5% of the sodium hydroxide for delignification. And as it was expected, we receive also very high mechanical tensile strengths without any reduction in the tensile strength for this sample, which is 0.5. But this was great. There is a problem here. We wanted to collaborate with the industry and commercialize this material. But the problem is that when you use only 0.5, I earlier mentioned that sodium hydroxide is being used in order to loosen the structure of our biomass. This wasn't enough to completely loosen the structure and break down the bonding between hemicellulose and lignin. What happened when in the next step of mechanical treatment through the high pressure homogenizer, it was so difficult to homogenize it because it was keep clogging the homogenizer. So our next step, when lignin gave a good property, we were thinking that how about use lignin? So we used lignin only, and we thought that we just using lignin, we can improve. But the problem with lignin was that lignin is hydrophobic and is not soluble in water. But we found that when we solubilized lignin in the alkali solution, which is the same pH that latex, natural rubber latex exists, we can make nanolignin that can significantly improve the mechanical properties of our latex without decreasing the tensile strength. Another example of the reinforcing polymer is the reinforcing thermoplastic polyurethane that we co-mill the cellulose nanocrystals with polyols, and then after drying that, you see that again in this case with very small amount of cellulose we can improve the toughness, improve the tensile strength without the compromise in the elongation. And one more example is when we wanted to add cellulose into the polyacrylonitrile as a precursor for carbon. We found that with addition of cellulose, we have a very higher D2G ratio, which is showing that graphite structure of the cellulose. What happens, the carbon, what happens when we add, when we add cellulose, we have a better carbon and at the carbon structure, and at a lower temperature, we are able to get the better types of carbon. So we also studied the effect of morphology of different type of cellular spinifex derived nanofiber, CNC. Still they are long, but they're shorter than CNF. And this is the cotton derived CNC. And we went through the wet spinning process for making carbon. So made white fiber, and after carbonizing that, what we found that addition of cellulose can help with improving the mechanical property of the carbon fiber, but specifically higher aspect ratio carbon works better. The error bar is huge. This is the problem of the unit that we use for making this carbon fiber is for larger scale, and then it was difficult to accommodate the smaller scale of our experiment. So another example of cellulose is on flexible electronics for potential ink application. So this is the work that my student Lauren worked on that. And this is basically, we use cellulose nanocrystal in this aspect. So the aim behind that, we wanted to make the conductive hydrogel for potential ink application. But the problem with the conductive hydrogel, it is difficult to get this synergistic high mechanical property as well as electrical conductivity at the same time. If you, for instance, especially we wanted to achieve the higher conductivity through using metal nanoparticles. If you use metal, they agglomerate, and then when they agglomerate, their mechanical property and electrical conductivity will decrease. So this was the motivation for us to work on that. But we use cellulose nanocrystal, and after the kind of surface modification, we introduced polymer block copolymer to the surface of cellulose. Basically, this is the first polymer, which is the PBAT, and then the second polymer, in this case we use PEG, was introduced on top of that. And what happens when we make this structure, then this can be hydrolyzed. PBAT can be hydrolyzed to PAA, which has the carboxylic acid group functionalities, and that can be a great precursor for the synthesis of metal nanoparticles. So to do that, our assumption is that what happened, then we have one system which contains polymer as well as the metal. So we avoid the agglomeration in the composite making through the shear force in the printing. Then these brushes can interact together and then give us the good properties. So the project wasn't that easy as we have expected. The problem is that there wasn't constant. What is important for us, retaining the structure of cellulose, retaining its high crystallinity in order to be able to do that. So the problem was that we have to find the method that is replicable as well as that consistent in order to be able to get same level of the degree of substitution just for the first very initial bromination. So we did the chemical vapor deposition. As you see that basically this is the freeze-dried cellulose nanocrystal. For the first step, we did this treatment in order to make a fine powder in order to have a uniform treatment. So there was a range of different kind of parameters that we have to optimize it in order to make sure that we retain the crystallinity and structure as much as possible. But we found that with increasing the degree of substitution, crystallinity decreased, so the fibers will be damaged. So we found that something around 10 to 11 is the best condition in order to introduce our polymer brushes. So successfully, you see that successfully we introduced PTBA, which is very hydrophobic. This is the sample which is just sitting on top of that, top of the water. And then we just along the way always characterize and analyze our samples. So then PEG also were introduced, which was approved by the different characterization method, the existence of PEG. So the hydrolyzing of the PTBA after introduction PEG also has been successful. We see here we retain the crystalline structure even after that. So the final stage is introduction of metal. We have two options: just add metal nanoparticles or synthesize that grow metal from the surface. So basically, the first approach doesn't give us control on the introduction of metal to the surface of polymer brush. The second approach, we choose the second approach to work with. And as you see that we made these kind of templates in order to basically one component of cellulose crystal and polymer brushes and metal nanoparticles. So we also use the same strategy in order to introduce iron oxide on the surface of the cellulose. This time is cellulose fiber. We know that iron oxide has lots of application, for instance, for the water treatment. But the problem is that it's not possible to make a membrane of the iron oxide because it's so brittle. So think about that. How about use cellulose as a template in order to synthesize these nanoparticles from the surface. And this is the image of the paper that we made from cellulose containing iron oxide. So you see here, this is the TM image of the cellulose fibers, and after a bit different loading of the iron oxide that we are able to control. The interesting thing is that when we have cellulose in the fiber, this is consistent. We are seeing that when you have cellulose in the system, the metal nanoparticles are much smaller, and also another things that the metal nanoparticles, they don't have tendency to agglomerate together in the system. They prefer to stick to the surface of the cellulose, which cellulose works as a capping agent and can be used instead of ranging different surfactants. So again, the we've done the, this is the super paramagnetic particles, and we've done the, and despite having really high content of the cellular seal, they have the sample which has the something around 60%, 40% cellulose, still have really high magnetic properties. So what we did, we used that in order to remove rhodamine B dye from the system through the advanced oxidation process, or iron oxides. Cellulose templated iron oxide works as a catalyst because they are very small, so they are very efficient. And then we have the oxidant in the system, which these two together, they generate the radical. But we found that in short time, we can remove 95% of rhodamine B from the system. And basically in this iron, the combination of iron oxide and the PMS oxidant that we use, they generate both hydroxyl as well as the sulfate radicals. So another example of the water treatment, this work has been done with Professor Benzau at the University of Stony Brook and his team. Basically, they took this is specifically with the Spinifex grass, they took the grass and then without any bleaching and lignification, they directly treated with nitric acid and sodium nitrate. That's the patented technology. And in order to introduce carboxylic acid group and negative charge, we know that heavy metals, they have positive charge, they get attracted to the negative charge. So through this way, we could remove 84% of cadmium from the water. And I have to mention that there is the best efficiency also obtained at the pH around neutral pH, pH 7. We know that at lower pH, we don't have high efficiency. So that's it. And I would like at the end to thank my group members, past and present, as well as the collaborator on this specific presentation. And I'm happy to take questions.
Thank you very much, Dr. Amiralian, for the interesting and inspiring presentation. Now we will enter the Q&A question. Ladies and gentlemen, if you have any question, you can raise your hand. Please. Okay, you. Please say your name and your institution.
Okay, thank you for the opportunity and thank you for the nice presentation. My name is Abdul Halim from International University of Semen, Indonesia. I have two questions. First question is for Spinifex derived nanocellulose, what method did you use to produce CNF? Tempo oxidized or kind of aqueous countercollision or any another methods? And the second question, during the lignification, lignin contain many alkalines. in your application, is the lignin directly used or need purification procedures? Thank you.
Yeah. Good to meet you. Thank you so much for your question. Actually, for Spinnavic, we didn't use Tempo, but I used Tempo for the sugarcane drive. But basically, very standard sodium, using the sodium sodium, not sodium borate, sodium chloride solution. That's the procedure that we follow, is established by Professor Izugai. So that's the very standard. Everyone is just using it. We are using that, but we are able to, it really depends on the application. We are able to tune the degree of oxidization in order to get the highest, which is around 1.5 millimole per gram, or I can say that around 22% to the lower if it's needed. About the lignin, you question, just making sure that I write your question correctly. You mentioned that when we retain lignin in the structure, we do any purification after alkali treatment. So basically what we can, because lignin is already exists in the structure of any plant. What we do in the delignin, we call it delignification, but basically these alkali treatment just loosen the structure, just breaks down the bonding between lignin and hemicellulose. Yes, we do have a black liquor at the end, so you can recycle that lignin and collect it, but we still retain some lignin in the structure, but it doesn't get detached from the structure of fiber with doing any mechanical treatment. I hope I answered your question. I understand it correctly.
Thank you, Dr. Amira, for your answer. Any other? Okay, Dr. Swasni.
Hi, good afternoon, Nasim. How are you? Hi.
It's great to hear old
Yeah.
colleagues.
Yeah, long time no see. Okay, Nasim, thank you for your presentation. Oh, very impressive. It's a long time, long time no see. And then I would like to have some question related to the development of composite between nanocellulose and other materials. Have you developed any other materials which is nanocomposite? Based on cellulose and, for example, metal oxide for water treatment.
Yeah, thank you so much. We don't do the I haven't done any kind of nanocomposite. There is a hydrogel. We did hydrogel with the carrageenan, but basically not for the not the composite. But as you see that we use it as it is for the water treatment. If you wanted to, for instance, you can add this into the electrospinning and make that membrane, mix it with the polymer and make the filtration for the water treatment. Definitely something is possible. Yeah.
Oh, okay. Yeah. Because in my lab, it's also, we have also research in the development of nanofiber. Yeah. Maybe we can, maybe later we can try to incorporate between cellulose and then nanofiber polymer.
Yeah, okay. Absolutely. I have to mention that I love to collaborate with people because I do believe that when we collaborate, we learn faster from each other and we can do more important work. And I'm sure that both Australia and Indonesia are very kind of supportive of collaborations. Any of you, anytime, I'm more than happy to collaborate. Yeah.
Okay. Yeah. Thank you very much, Nasim.
No worries.
Good luck for your presentation. Okay. See you.
Thank you. Thank you very much, Dr. Amira. And okay, you please say your name and your institution.
Well, thank you very much, Dr. Nasim, for the interest talk. I'm Nia from Research Center for Environment and Clean Technology. I would like to ask about you explain about the nanocellulose from non-wood materials. So when you talk about flexible electronics, what first pops up in my mind is about the stability of the material. So did you conduct a study about the stability of the materials when comparing to the existing materials, especially in the application for flexible electronics? How the stability of the cellulose nanofiber based hold up over time, and considering its organic nature, and in your opinion, what's the factors contribute to the long-term structural integrity of these materials? Thank you.
Yeah, thank you so much. That's a really good question. So you know that this is a general term for us, general idea that we are, because the cellulose, I have this conversation with another colleague today, that using cellulose in the building material, termites gonna come and attack it. But basically, if we look at our ancient work, ages ago, million years ago, cellulose in the form of rice husk or straw is being used mixed with mud for making building. But same principle applies here as well. For instance, you see that a lot of these batteries, they have the cellulose-based paper as a battery separator. So this is very similar concept applies for the long term. But you know, the main reason that we wanted to work on flexible electronics, we have the electronic waste is huge. So they are when we look at the stats, we see that nowadays people keep changing their mobile phone and everything. And honestly, they don't live too long. Even after 2 years, you see that your mobile phone's performance significantly decreased. But my answer is that when we made this, we haven't used this in application yet. But when we made these, hydrogel is very stable, it doesn't settle, and then we monitor the properties of the metal as well as the hydrogel for few months, still is stable. So when they become in the dry form, they are just more. And I have to mention that as well, when you dry it, basically you cover, it really depends on the number of brushes, you cover the cellulose with all other polymers, which is not affected. Mainly cellulose can be used as a template for the formation of this system.
Doctor, the presentation is closed now, and the committee want to give you a certificate and take a picture.
Sorry, I have one question from another friend. Yolin asked me, can I answer this one as well?
Yes.
In the team.
Yes.
Hi, Yolin. Great to see you here. Yes, twin screw. Yolin asked that for this nanocellulose production using twin screw technology. Yes, that can be used. We've done some work. Actually, we published one book comparing different methods, twin screw and high energy milling and high energy high pressure homogenizer. So it really depends on which ultimate application you wanted to use it. If you want a high degree of refining and having very nice individual fibers, for instance, for our latex project, twin screw doesn't work because it gives us mainly the bundles of fiber, which is coarser and bigger fiber. That could be good for other type of, for instance, packaging material, some kind of fiber-based packaging material. But yes, definitely is possible. It's cheaper, much cheaper in the processing, but depends on what is the ultimate application.
Okay, thank you, Rahim.
Thanks.
You.
Thanks.
And the organizing committee want give you the certificate and take picture. Thank you, Dr. Amira. See you
Thank you so much. Thanks. It's my great pleasure. I appreciate it.
Bye.
Bye.
Thank you.
Thanks.
Okay, next we will invite the second keynote speakers, Professor Ji Chen Liu, talk about fertilizer industry waste. Prof. Liu, are you ready? Ready? Prof. Lu?
Yes, I'm ready.
Okay, thank you. First of all, allow me to read your CV. Professor Ji Chen Lu is an accomplished environmental engineer and academic leader. He holds a PhD in environmental engineering from the University of Delaware and has been serving as a professor in Department of Chemical Engineering at Taiwan Tech since 1990. Throughout his career, he has made significant contribution in the field of physical chemical processes for wastewater treatment and reuse, as well as the extraction and recovery of rare metals from electronic waste. Professor Shaw has held various leadership positions, including Dean of the College of Engineering and currently as Vice President at Taiwan Tech. Microwave assisted leaching of rare earth element from phosphogypsum will present by Professor Ji Lu. Okay, Professor, are you ready? Time is yours.
Okay, good morning. Just a minute, let me get my slide so that I can share with everyone, okay? Because all of a sudden I lost my content. Can you see my slide now? I'm sorry, can you hear me? Okay, so I'm honored to be able to give a talk in this conference, and I'm grateful for the invitation from chair of this conference, Dr. Athanasia Amanda Septavani. Okay, I'm JC Liu from Taiwan Tech, and I'm going to talk about microwave-assisted leaching of rare earth elements from phosphogypsum. Why rare earth elements? For people like me who major in chemical engineering since my undergraduate days, okay, rare earth elements stand for 17 elements that lie somewhere in the periodic table, okay. However, for the past 30 years, people have come to realize that these rare earth elements, actually, they play a very key role because they are essential for all sorts of high tech, such as renewable energy, electric vehicles, medical, cell phone, computer, military defense, and industrial catalyst. That's why they got the name, the vitamin for high tech. And another name is like people call rare earth elements as the gold of the 21st century. From this table, you have some idea about the production of magnet that are mostly used in wind power, as well as in medical instrument that require rare earth elements. And on the x-axis, we come to realize that the demand for EVs and for wind turbines actually keep increasing throughout the years. For your information, this table shows the world's rare earth elements production and reserves. As we may find out from this table, the 4 countries, China, Vietnam, Brazil, and Russia, stand for the 4 countries that own the most reserves, okay, with the number 1 as China. However, in terms of production, China every year produces about 168,000 metric tons, which is the number 1 for the whole world. Okay. On the other hand, the second and the third producer for rare earth elements are United States and Australia. But somehow the reserves in these 2 countries are not comparable to the 4 countries on the top. That's why a paper published by United States Geological Survey with the title, "The Future of Rare Earth Elements. Will these high-tech industry elements continue in short supply?" that calls for our attention to the shortage or the stable demand and supply for rare earth elements. That's why people turn our attention to the secondary sources, namely all sorts of industrial waste. But from this table, we notice that actually phosphogypsum stands for the most promising one, because every year the global production for phosphogypsum reaches almost like 2 to 3 million metric tons. And the average IE content, as revealed from this table, actually is also comparable to those of nickel metal hydride batteries or polishing powders. Why phosphogypsums? Actually, phosphogypsum is the main waste or byproduct from fertilizer production. Okay, as we may find out from this table, once we use the wet chemical methods to produce phosphoric acid, in the meantime, we also find some calcium sulfate precipitate with crystalline water ranging from 0, 0.5, and 2, okay, namely anhydride, calcium sulfate hemihydrate, and gypsum. And this is the so-called phosphogypsum. Namely, mainly it's gypsum, but it contains certain amount of phosphorus. Okay, so typically the content of phosphogypsum, you find calcium, sulfur, phosphorus. But in the meantime, as pointed out from this table, REE, okay, could constitute 0.01 up to 0.4%. Okay, and the total amount of phosphogypsum in storage by 2020 already exceeded 600 million tons worldwide. In the meantime, the annual production was about 300 million tons. However, only 15% is recycled and reused. Mostly, namely 85% phosphogypsum is dumped without any treatment, and that could cause environmental problems. For example, leachate could leach out radionuclides, heavy metals, and contaminate the receiving water bodies. In the meantime, the dust and some aerosol could produce some atmospheric pollutions. Okay. And in terms of valorization of PG, okay, they can be applied in building. Why? Why not? Because it's mainly gypsum. It could be applied in agriculture as a amendment because it contains phosphorus. And it could also be reprocessed and applied in pollution prevention. For example, carbon dioxide absorption and fixation. It could also apply in industrial sectors. However, now people turn our attention to the PG as a promising source for raw materials, such as IE and phosphorus. Why only 15% recycle and reuse rate? As pointed out by researchers, it's because of the high content of impurities, especially phosphorus, because with the 3 to 4% phosphorus, that could increases the setting time and also decrease the mechanical strength when utilized as construction materials. Okay, and this figure shows you 3 different crystal structure for calcium sulfate as commonly found in the PG. Okay. And according to some study, okay, there are 2 main existing forms for phosphorus and rare earth elements existing in the PG. One is those become absorbed onto the surface of PG. And this fraction stands for the fraction that are easily leached out. On the other hand, we also find some IE and phosphorus existing mainly through isomorphous substitution with sulfate groups. So they become part of the crystal matrix. And this part actually is known to be difficult to be leached out. Okay. Luckily and gratefully, we got the PG sample from Petrokemia Gresik, a state-owned fertilizer company in Gresik, East Java. As pointed out by this website, Petrokemia Gresik just opened a new production line for phosphoric acid in 2014, okay, with a total budget of $200 million, and production capacity of 200,000 tons. Okay. In the meantime, they also upgraded the existing old manufacturing line that stands for another 200,000 tons phosphoric acid production. Okay. And we were honored to be able to have the opportunity to visit Petrokemia Gresik last year and have a very inspiring talk with the plant managers and engineers in the company. Okay. Once we got the PG sample at the beginning, we tried to understand the crystal structure. We found gypsum, bentonite, which is the calcium sulfate hemihydride, and hydride as well as quartz. Okay. And from the XRF preliminary analysis, we also find some rare earth elements, namely yttrium, erbium, neodymium, GD, cerium, and samarium. Okay. And this slide shows you the SEM and EDEX analysis of our sample. Okay. And by aquaregia digestion, we noticed that mainly there are 4 types of rare earth elements: yttrium, lanthanum, cerium, and neodymium, with total concentration of 279 milligram per liter. Okay. And this figure shows you the residue after subject to aquaregia digestion. It implies that all the phosphogypsum would dissolve except a inert quartz. From literature, we understand that hydrometallurgy have been widely used for IE recovery from all types of minerals or waste. Okay. It's the mainstream in recent years that owns advantages of, say, low energy input, high purity, and also relatively low air pollutant emissions as compared with the other pyrometallurgy. Okay. However, the shortcomings of hydrometallurgy is that it requires concentrated acid, and as a result of producing huge amount of waste acid. In the meantime, it takes a long reaction time unless at elevated temperature. Okay, so in this study, we use conventional extraction, where you find a water-based shaking bath, and the samples after subject to extraction go through solid liquid separations, and then the residual is subject to surface analysis, whereas the liquid is subject to ICP OES analysis. Okay. And for study before, okay, last year, we use a technology called subcritical water extraction, what is subcritical water? It stands for water with temperature ranging from 100 to 374 degrees C, and normally with pressure higher than 10 bars. Okay, when under subcritical condition, we found decreased dielectric constant and surface tension of water. Okay, so it proves that under subcritical water condition, the mass transfer efficiency will be enhanced. Thus, subcritical water extraction technology has been applied for biorefinery as well as valuable metal recovery from e-waste. Okay, and this is the apparatus we use in our former study. Okay, basically, this is a heated, pressurized hot water. Okay, so it's a simple lab scale equipment. Okay, I'm not going to the detail for our previous study using subcritical water, but from this table, I'm sure you noticed that when comparing results from subcritical water extraction with the conventional extraction, notably the leaching efficiency is much better. Okay, in the meantime, the total energy consumption is almost only 1/3 more efficient, and the so it means that subcritical water extraction could be competitive. Okay, so this is the summary of our previous study. Okay, and we published the results. Okay, but today I'm going to talk about a new topic, and the graduate student Laurentia, who was responsible for this study, was graduated and now working in Taiwan. And the topic is recovery of rare earth elements and phosphorus from PG using microwave assisted leaching. Okay, so these are the objectives. We want to use microwave assisted leaching, and the target elements include yttrium, lanthanum, neodymium, and phosphorus. Okay, we use microwave. In the meantime, we will look into the addition of hydrogen peroxide, because the combination of microwave and hydrogen peroxide is a typical AOP, advanced oxidation processes, that could somehow facilitate solid dissolution. Okay, we will also study the feasibility of selective precipitation of phosphorus as struvite, namely magnesium ammonium phosphate, a fertilizer that could be reused right away. Okay, so this is the equipment that we use. Okay, we put samples in acid solution subject to microwave assisted extraction reactor. Okay, and followed by solid liquid separation and analysis. This is a photo, close-up photo that shows you the microwave assisted leaching system. Okay, basically the samples is placed inside the Teflon vessel, and it could be screwed tight. That is to say, when heated, the temperature could be higher than 100 degrees C, and the pressure could be higher than 10. And somehow the condition within the microwave system will be more or less similar to the conditions in subcritical water conditions. Meanwhile, this system could be easy to control the temperature, the heating rate, as well as the mixing speed. Okay, why going microwave? People know that microwave could save solvent usage. It could enhance selectivity, and normally we find higher yield in relatively shorter time. And microwave is known as green because it's energy efficient. Okay, it's been applied for spent automotive catalyst, spent lithium-ion batteries for the recovery of platinum group metals, lithium, and cobalt. Okay, and for the combination of microwave and hydrogen peroxide, actually, it's been applied for rare earth elements recovery from spent CRT phosphor and spent FCC catalyst. But I like to point out that the temperature is lower than 100 degrees C. That is to say, it's not still subcritical water yet. It's only heated water and accelerated reaction only, okay, which is different from what we are going to look into. Okay, so I want to show you the results. First, we compare three types of acid, okay, hydrochloric acid, nitric acid, and sulfuric acid. We found that hydrochloric acid could yield the highest leaching efficiency, normally higher than 60%, okay, when using very dilute concentration, only 0.5 norm, okay, and at 20 gram per liter solid concentration, 100 degrees C, and the reaction time could be as short as 5 minutes, okay. And when using hydrochloric acid and changing the concentration, we find that the leaching efficiency increases with increasing hydrochloric acid concentration. And we could find almost complete dissolution of rare earth elements of yttrium, lanthanum, and neodymium. And even for phosphorus, we could leach out up to 87% of phosphorus. Okay. And as expected, the leaching efficiency decreases as solid concentration increases. It's because there could be inadequate acidity available. Okay. We also examine the temperature. Somehow the results are unexpected because normally people will think that the higher temperature, the higher the leaching efficiency. But somehow we notice that temperature is only an insignificant factor here. Why? Why so? Okay. We try to come up with some explanation. It's complicated, not as straightforward. For example, we notice that when under high temperature, high pressure, okay, gypsum will be converted to anhydride. However, the solubility of anhydride will decrease as temperature increases. Nevertheless, when increasing pressure, the solubility of anhydride on the contrary also increased. That is to say, we find contradictory force acting in different directions. And we also know that the mass transfer efficiency is enhanced by microwave. Perhaps we hypothesize that that's somehow we don't find temperature as a determining parameter. We also assess the energy requirement when applied for IEE and phosphorus recovery from PG. Okay, that again proved that microwave is very efficient when it comes to energy consumption. Okay. Finally, we also study the effect of hydrogen peroxide. This is also unexpected, okay, because as we increase hydrogen peroxide concentration, the leaching efficiency of P, phosphorus, increased. However, those for IEEs decrease as hydrogen peroxide concentration increased. Okay, why so? Okay, this is different from our expected, what we originally expected. Okay, so we try to add another experiment, trying to analyze the leaching of zirconia. Okay, we noticed that as hydrogen peroxide concentration increased, okay, the leaching efficiency of zirconia, which is a impurity, also decreased. Okay, so and we also detected and analyzed by XPS to confirm the formation of zirconium dioxide on the surface of PG after subject to microwave-assisted leaching with 1% hydrogen peroxide. And that's why we propose that it's because when under the influence of hydrogen peroxide, a very thin layer of zirconium dioxide is formed on the PG surface and somehow hinder the dissolution and leaching of rare earth elements. Finally, we also tried to see if we could carry out selective precipitation of P in the acidic extractant by adding magnesium chloride to induce the production of magnesium ammonium phosphate. This is a simulation results at molar ratio of magnesium to ammonium to phosphate at 1 to 1 to 1. Unfortunately, we cannot find any formation of magnesium ammonium sulfate. We can only find trace amount of hydroxyapatite form. Okay, most of the time, the dominant solid will be still gypsum and a little bit of brucite, which is magnesium oxide. Okay, so I'd like to summarize our main findings from applying microwave assisted leaching for PG. Okay, first one is when using 1 norm hydrochloric acid of 20 gram per liter solid concentration, 100 degrees C in 5 minutes. The leaching efficiency of yttrium, lanthanum, neodymium, and P was 106, 101, 99.7, and 87% respectively, with low energy consumption. And addition of hydrogen peroxide enhanced the leaching efficiency of phosphorus only, but inhibited leaching of IE due to the chemical preservation of zirconium dioxide. Okay, and selective precipitation is not feasible due to the interference of calcium. So on the recovery of rare earth elements from PG, okay, I'd like to point out the process intensification, such as critical water extraction and microwave assisted leaching, were effective in leaching out yttrium, lanthanum, and neodymium. Both were energy efficient. And we like to point out that further concentration and purification as well as scale up are required in the further study. Okay. Finally, I'd like to acknowledge the participation and contribution of Professor Wamadewanti of ITS and Laurentia, our former graduate student from Sidoarjo, Indonesia, who after receiving her master's degree has been working in Taiwan for her first year stay. Thank you very much. Thank you.
Thank you very much, Prof. Lu, for your nice presentation. Now we go to ask and question. Q&A. From audience, if you have some question, you can raise your hand. First, say your name and your institution. You? No question? Yes. Please.
Thank you. My name is Hans from Parahyangan Catholic University. My question, there are 2 questions. First, how about the scaling up for the microwave reactor for extraction? As we know that from the lab scale to maybe industrial scale, there are some challenges. And for the second question is regarding the selectivity for the metal extraction, because sometimes we have crude extract with various metals. How can we selectively extract the metal, or as you have presented, the selective precipitation might be a better strategy? Thank you.
Thank you very much, Hans from Parahyangan University. You just had 2 very excellent questions. First is about the scale up of microwave reactors. That's the major disadvantage of any microwave reactors, because scale up is always a challenge, and I must say a difficult problem as related to microwave. But from literature, I come to realize that in Japan, we already find some pilot scale microwave reactors. So perhaps that could provide us a hope. Okay. And the second is about the selective leaching of metals from any waste or any solid. That is also a very good question. But allow me to say that now the mainstream technology or approach is like perhaps we try to leach out all the metals from the solid, okay, then followed by selective precipitation. For example, as we know that in the acidic extracted from PG, we find yttrium, lanthanum, and neodymium, together with aluminum, iron, and perhaps some heavy metals. How could we do? Usually we could do by adding oxalate, because oxalate is known to be able to form specific precipitation with all sorts of rare earth elements. And oxalate as a selective precipitation reagent has been widely applied even for industry. So this is for your reference. But thank you very much for your very good questions.
Thank you very much for your answer. Next audience. Are you have a question? Okay, please say your name and your institution.
Kalau pakai PDF gimana nanti apply-nya?
Hello, tes. My name is Karelius. My institution is Institut Teknologi 10 November Surabaya. My question is, in your conclusion or summaries, why some of the rare earth extraction yield are more than 100%? Can you explain how you determine it? Thank you.
Thank you for the very challenging questions from our young friends from ITS, where I find many good and old friends in ITS. Okay. Yeah. Also, I challenge myself about why we find 106%, 103% recovery or leaching efficiency for yttrium and for lanthanum. Okay, my answer is this: because we are chemical engineers, we are used to working with 99% purity solvent chemicals. But bear in mind, we are dealing with waste, okay, which is heterogeneous. This batch is different from the next batch, okay, and even from this batch, each grab from the sample bottle, okay, container will be different, okay. But what I can tell you is in our lab, we always ask students to conduct experiment in triplicate, so that at least we have some idea about the quality of the experimental data. So in a way, I would say 106 may be a bit too high, because my personal criteria will be always for any wet chemical experiment, 5% plus minus is acceptable. Okay. Do I answer your questions?
Thank you, Prof. Lu. Another audience? You have question again? We still have 2 minutes for the question. Okay, I will ask you, Professor Lu, about your research. How much can you extract the rare earth element from the phosphogypsum? Yeah, the first. The second is the research, the method for research is by microwave. Why you use microwave? Because I think for scale up, maybe you have problem. Thank you.
Thank you very much, dear moderator, Dr. Desak. Okay. For the second question about why choosing microwave, I already answered Hans from Parahyangan. Like, yes, I do agree with your comments that microwave reactor is difficult to be scaled up. Okay, but as I pointed out that in Japan, according to some literature, already we find scale up microwave reactors. So perhaps that could provide as a breakthrough or as a hope. Okay, but I must agree that the microwave has its own shortcomings. The second question is how much rare earth elements that we could extract out of the PG. Okay, this is also a very good question. I must point out that the total rare earth elements content in the system, which is about less than 300 milligram per kilogram. That is to say, more or less 287 milligram per kilogram. Okay, so even if we could reach out 100%, 106%, okay, the concentration of REE in the acidic extraction is still low. So that's why I listed in my final conclusions that we need further concentration and purification. For purification, as I pointed out, we could add oxalate to separate rare earth elements from other heavy metals. However, the extractant still has to go through concentration by using ion exchange resin, by using membrane, so that it could reach the concentration high enough to subject to possible selective precipitation by adding oxalate. That's my opinion. Okay, but thank you for the two very good questions. You got me.
Okay. Okay, you're welcome, Prof. Lu. Now, the end of the session, the organizing committee will give you a certificate and take picture. Please applause for him. Okay, Prof. Lu, see you.
Yeah, thank you. Thank you very much. Thank you.
Thank you very much. You're welcome. Ladies and gentlemen, the audience, now we will present the third keynote speaker from PT BRK Niaga Medica. Mr. Ferdi Ferdian Kusnadi is already here. And okay, before you start, let me read your CV, Mr. Ferdi. Mr. Ferdi Ferdian Kusnadi, STP, is a seasonal professional with a wealth of experience in the field of industrial chemistry, laboratory management, and quality assurance. Ferdi's career journey is marked by a dedication to advance the field of industrial chemistry and laboratory practices. He has held a variety of key positions, showcasing his expertise and commitment. Ferdi has been a registered assessor of competency for industrial chemistry since 2019, demonstrating his proficiency in the field and served as a secretary at the Indonesian Food Laboratory Association, known as LPI, from August 2012 to 2018. This role allowed him to contribute to the advancement of food testing and analysis in Indonesia. Ferdi has been actively involved in the field of chromatography and mass spectrometry since 2003, showcasing his enduring commitment to cutting-edge analytical technique, played a pivotal role as a co-founder and member of the Indonesian Food Laboratory Association, ILPI, contributing to the growth and development of association from 2012 to 2018. Ferdian Kusnadi extensive professional background, educational achievement, and active involvement in the field of industrial chemistry make him a respected figure in the industry and valuable contributor to the advancement of laboratory practices in Indonesia. GCMS technology for dioxin and furan analysis will present by Mr. Kusnadi. Mr. Kusnadi, time is yours.
Thank you, Ibu. Good morning, ladies and gentlemen. Thank you very much for the time for us to be present here to share about the latest solution available for dioxin analysis. So before your lunchtime, hopefully you still have enough energy to concentrate to the last topic before lunch. So talking about the dioxin, actually is a very common pollutant that we all know this is very extremely toxic compound. Dioxin coming from the environmental, from the industrial activity mainly. So because it's exposed to the environmental, dioxin can be involved in our food chains, as well as in the air, in the water, in the soil. And because this compound is very toxic, so we have to be very careful. And to analyze this dioxin is very challenging analytical techniques. Why? Because dioxin is consists of a lot of isomers as well as congeners. What is the difference of isomers and congeners? Isomers, as we all know, that we have similar compound with the same molecular mass, for example, but different structure. That is isomer. But congener is not only similar in the structure and molecular weight, but also similar in the properties. So it's very difficult to separate, and because the concentration is very, very low, so it's also difficult to detect accurately. So I would like to share the latest technology available to analyze this dioxin. So what happened about the existing method, talking about the dioxin analysis in the environmental sample? Mainly we refer to the US EPA method 1613B, which is in this official method, we use the high resolution mass spectrometer to detect and analyze the dioxin samples. But currently, should we break for the, excuse me, so we have to stop because the azan. Okay, thank you very much. So we continue the presentation. Regarding the official method currently for the dioxin analysis, the most common reference is using the US EPA 1613B. So in this method, actually, they're using the high resolution mass spec to analyze the dioxins and furan compounds. But recently, SGS Axis actually developed the advanced method to analyze dioxin and furan using alternative technology, which is GC, MS, triple, quadruple. Why is it possible now? So we will explain the next feature. Okay. Because now we can use triple quad mass spec to analyze dioxin, so it is a good news actually for all of us, which is the researcher in the laboratory, for example, because talking about the GC-MS triple quad, it's very common instrument in the lab, not as difficult as high resolution mass spec in terms of maintenance as well as the operation of the instrument. And of course, the initial price of the instrument also very much different compared to the triple quad. So when we use the triple quad, it is very common, and a lot of people already use the triple quad for other application as well, not only for dioxin. For example, in the world of the pesticide analysis, actually very common customer in the laboratory using the GC-MS triple quad as well. So not easy to use. Even the user of single quad GC-MS can easily use the triple quad GC-MS, just because of the mode of operation is different. In the single quad, we use mainly SIM or scan mode, but in triple quad, we commonly use the multiple reaction monitoring or MRM mode actually. That is the condition. Okay, why originally in the beginning of the dioxin issue, the requirement must use the high resolution mass spec? Because we know the most toxic dioxin compound is 2378 TCDD, tetrachlorodibenzoparadioxin. But in the fact of the sample, actually, a lot of similar compound to this particular very toxic compound. So how we ensure that our analysis can be detect this very specific compound with other things actually inside the sample. So we need very good separation as well as very good sensitivity to detect. Okay, dioxin structure is like this on the left hand side. The most toxic one is on the left hand side, 2378 TCDD. And also in the dioxin analysis, commonly we also analyze furan on the right hand side, which is the compound is similar. And in the PCDD itself, we can see this, we have total 75 isomers of PCDD. And PCDF, we have 135 isomers. So this is actually the main challenge, how to identify accurately our target compound. So in the dioxin, also another challenge is not about the sensitivity, but also the requirement itself. To detect dioxin, actually, we need to be able to calculate the sample toxicity. If we are familiar with the testing in the laboratory, mainly we do the quantitative or qualitative analysis, right? For quantitative analysis, we just need to analyze how much the compound inside our sample. How low? How is the concentration? What is the concentration? But in the dioxin analysis, it's not enough. The concentration report is not enough. What we need to report based on the official method, we need to report the sample toxicity. How to get the sample toxicity? we need to sum all these toxic PCDD, for example, that every single compound actually have very unique TEF, toxic equivalent factor. For example, we can see number 1, TCDD, 2378 tetra CDD, the TEF number is 1. Mean to say that this is the most toxic compound. Similar with the penta CDD, 1. But how about hexa CDD? Is only 0.1. So even though the hexa CDD have concentration same with the tetra CDD, the toxicity level is less by tenfold. At the end, we need to calculate this as the summation, the total summation of the concentration of each PCDD multiplied by every single toxicity equivalent factor become the sample toxicity. So to do this, actually, we need special reporting tools, because not only to sum this data, we need also to check other parameters like the recovery and also the internal standard response factor. So a lot of parameters need to be checked actually. But don't worry, because now we can use the GCMS triple quad, and also there is proven technology for the automated sample preparation. So we need to be worried. Actually, we can use the complete set of the sample preparation as well as the GCMS triple quad to analyze dioxin in the environmental sample as well as in the food samples. The basic method of sample preparation, beginning from sampling, of course, it depends on our sample. It can be water, soil, or air, or maybe food sample, also possible. However, we need to change this form of sample into the compatible form of the solution that can be injected to our GCMS. The main step of preparation consists of extraction and also the cleanup. And these two steps can be done automatically with the automated sample preparation apparatus. Okay, why we can use GCMS now? The official method of US EPA is basically result-based method. What does it mean? So it's not very specified. We use the specific method or instrument, but there is a lot of requirement to be fulfilled to be able to use the method that we choose. For example, there is requirement of the separation for dioxin, that the separation of the key toxic compound, which is the 2378 TCDD, must be less than 25% of the value. Okay? And high-res GCMS, of course, very easy, can meet this requirement. But we can see on the top of the graphic here, now the GCMS also can perform the similar result, which is less than 25% value of the separation from the very toxic compound. So this is one criteria, the separation. So we can use the GCMS to fulfill this requirement. And also if we see the hexa CDD, we can see the separation is very good. Even this is can be say that baseline to baseline separation, right? Very good separation. And beside the separation requirement, also there is the requirement of the sensitivity, of course. Why GCMS can do the job? Because now the technology is enhancing actually. The GCMS sensitivity now can detect compound up to femtogram level, which is very, very low amount of substances can be detected by this instrument. We can imagine femtogram, it's how much femtogram? 10 the power of minus how much? Minus 15, yeah. Minus 15 gram. So it's very, very low amount of substances that we can detect with this instrument. Okay, so if we compare the result, the method, this both method, EPA 1613B compared to the triple quad method, we can comply to all this requirement actually. Valid for EPA compliance reporting, yes, because we do the reporting according to the EPA requirement. Matrix validated, yes, same as 1613B. Sample preparation, same. Method detection level, yes, we can do very low. The requirement is 4.4 picograms. Picogram is 10 the power of minus 12, right? Gram. But our instrument can detect up to femtogram, even lower. The requirement is only 4.4, but the actual performance of the triple quad, we can detect up to 1.1 picogram per liter. Detector, of course, different because different technology. And the tuning requirement, the GCMS triple quad is much more easier because we can use the standard tuning method in the GCMS MS, no special requirement. Logmas reference requirement, we can use the infusion of PFTBA to make sure the performance of the instrument. And the reporting units also the same. We can report the toxicity equivalent, TEQ. So this is just to inform you the equipment, the GCMS MS. It looks like ordinary GCMS, right? Very similar. The difference is only the MS part on the left-hand side here. It's different. And the separation of the mass is 2 times the ionization. In the GCMS MS, we ionize the sample 2 times, once in the sample ion source, and then we can select the parent ion, and then we can break down this parent ion in the collision cell into the product ions. So at the end, we can detect the product ions and also the parent ions. And this one is the picture of the sample preparation equipment for the dioxin sample. The basic principle of this equipment is basically the pressurized liquid extraction and cleanup, following up by the cleanup step. Okay, this is just to show you the configuration of the instrument. We already have the method ready to use, the hardware configuration, the instrument condition, as well as the reporting template to make you easy to report the result of the dioxin analysis. Okay. So we now use very simple equipment like this, only benchtop instrument, compared to the very big high-res instrument, magnetic sector. And we have resources actually available on the website. You can try to Googling through the website, the application note or the technical notes related to the dioxin analysis easily, and you can download it for free. And also we have the comparison about the result, comparison of high resolution mass spec with the GCMS MS for each compound, the dioxin compound, and the result is very comparable. Okay, for more information, if you are interested to find, you can anytime contact us from BNM, or you can go to our website and find a lot of resources actually available on the website. Thank you very much for your attention. And if you have any question, please, thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Mr. Kusnadi. You can sit here. Okay, we have still 5 minutes for Q&A. For the audience, if you have question, you can ask your name and your institution. Okay, you. Please.
Okay, thank you for the nice presentation. I have 2 questions. Actually, I'm Abdul Halim from UIC, Universitas International Semen Indonesia. I have two questions. Actually, I never work in GCMS, so maybe my question is really basics question. My first question is, it's the GCMS instrument what kind of sample the GCMS instrument can imply? For example, is only powder or seed or liquid or any kind of samples? It's the first question. And the second question, it's the product is imported or created or produced by the BNM. Thank you.
Okay, thank you for the question, Pak. You have two question, very good question. How we can use the GCMS? The first question, how we can use the GCMS? Is it for only liquid or semi-solid or solid? The answer is the limitation of the GCMS is actually designed to analyze organic compound which is volatile and stable in the high temperature. That is the common scope of the GCMS applicable actually. Okay, but in fact, we can also analyze the solid sample with GCMS by doing sample preparation. And it's very common. For example, we analyze the fish sample for the dioxin. Fish is a solid, right? Solid sample. How we analyze dioxin? Because dioxin is GCMS compound compatible, so we can extract the dioxin from the fish. Also, in other world, actually, we can analyze the narcotics from hair, actually, which is used in the forensic laboratory to check the compound inside the hair, which drugs actually this person consumed before. Even the sample is liquid, we can inject directly if it is solvent-based. If it's water-based, we need to extract. And another situation, we have non-volatile compound, but we only have GCMS in our lab. What to do? We still can use our knowledge of the sample preparation to change the form of the compounds into the volatile one. For example, we can do the esterification of the organic compound, become ester compound of its compound, so it's become GCMS compatible. So basically, I'm one of the fans of the GCMS equipment actually, because GCMS is very established technology and the library itself is already established. There is a lot of database. We can identify compound, unknown compound, and also we can do also non-volatile if we have the derivatization method. So that is the answer. And for the second question, of course, until now, we still import this product from other countries. In this case, Agilent is a US-made instrument actually, but the factory we have in Germany, in US, in Japan, in Singapore itself, we still have many sites. Okay, Pak, hopefully can answer your question. Thank you.
Okay, thank you, Mr. Kusnadi. Another question? Okay, Dr. Swasmi.
Okay, thank you, Pak Kusnadi. I would like to ask whether this equipment can measure or analyze the PFAS per fluoroalkyl substance. Thank you. And maybe is there any method that has been developed? Thank you.
Thank you very much. This is very important issue in the environmental world, ya Bu ya. PFAS. Everyone talking about PFAS, but because my topic is dioxin, I don't touch about the PFAS, unfortunately. But talking about PFAS, for sure we have solution for PFAS analysis, but unfortunately it's mainly LC-MS based. Mainly, but some PFAS, because when we talk about PFAS, per and polyfluorinated organic substances, is talking about a lot of compounds, right? Hundreds of compounds actually categorized as PFAS. So some PFAS is a GCMS compatible, and some others is a LC-MS compatible. So ideally, if we have both, we can analyze all PFAS. Basically, yeah. Yes, maybe we can follow up to provide the application note to Ibu Doctor, actually. For the PFAS analysis, yes. Yeah.
Okay, thank you very much, Mr. Kusnadi, for your answer. Because the time is over, the organizing committee will give you the certificate and souvenir.
Mr. Freddy, I apologize. Maybe you might want to stay for a while because we will have photo session together with Miss Amanda. Please.
Please come forward and
Aku kira 12.20, makanya aku duduk di belakang dulu. Lunch break aja.
langsung lunch break ya.
Lunch break sampai jam 13.20.
13.20.
Ini aku ingetin.
Ladies and gentlemen, this is the end of the keynote speaker session. And after this, we have to take a rest until 13. And I give the MC to the announcement.
Thank you very much, Budusak, for moderating this keynote session. So I would like to announce that we will have a lunch break for 1 hour and 20 minutes. So we'll gather again in this parallel room on 1:20 p.m. Okay.
Yeah, ladies and gentlemen, the lunch will be served at the building in front of us. And for all oral participants, don't forget to collect your PPT and don't forget to find your own room from room A, C, D, F, or stay in the main hall. So pay attention to the program book. So
Okay. Enjoy the lunch and we will see you again in the oral session this afternoon. Thank you very much.
thank you very much.
Kira 11.20 makanya tadi.